Welcome to SciShow Tangents, the lightly competitive knowledge showcase starring some of the geniuses that make the YouTube series SciShow happen this week. Taking my place on the science couch is a thing that is not me, a very special guest. It's a Trace Dominguez.
Hello.
Trace is of the YouTube series Uno Dos of Trace, which you can find at youtube.com/Trace Dominguez. Can you spell your name for me?
It is TRACE and then DOMINGUEZ.
Thank you for coming. I've been watching you on YouTube for many years.
Thanks. Yeah, this is exciting. I've never tangented with y'all. I'm excited to go on tangents.
To make a thing with you.
Yay.
Yeah. Trace, what's your tagline?
Stickers are great.
Glad you like did we provide you with a sticker?
I got a SciShow sticker and I'm really excited about it.
We have also SciShow Tangent stickers that are available at
DFTBA.com. I've got one of those.
Designed by Hiroka Matsushima. And we're also joined on the Science Couch by Ceri Riley. Hello, Ceri.
Hello, I'm here too.
How are you feeling about today's topic?
Oh, I'm excited. I'm a little bit nervous. This is something that I paid a lot of money to get a piece of paper to say that I know it. So
you better know it.
I better know it.
Ceri, what's your tagline?
Just a floating head.
Oh, yeah.
Which is a visual joke.
Ceri's shirt is exactly the same color as the couch, so we are all a little bit thrown off.
Just imagine what color the couch is. Pick your favorite color.
Everyone knows. It's canon. The Science Couch is yellow.
Is it yellow?
If they're true HFS fans from back in the day, they know what color the couch is.
Sam also matches the couch that he's on.
A little bit. True. He's sort of not a floating head. He's like partially invisible.
Yeah, like Predator camo.
Yeah. Mm-hmm. You need some leaves on the couch, too. And then put right in.
Sam Schultz is also here. Hi, Sam.
Hello.
What are you working on these days?
I'm working on the SciShow Science Kits. Universe Unboxed is what they're called. Ooh. Yeah, and you can order them online. universeunboxed.com.
Boom.
I believe.
That's so exciting. Yeah, and there's videos that go along with it, so you can see how the experiments work.
Yeah, you can see Hank getting goofy in a lab coat.
It's been really fun.
Yeah.
And Stefan is, what's your tagline?
Just a floating butt.
Together we make a human.
Yeah. Yep. No, a butt and a head together does not make a human.
Yeah, you connect them. There's two holes.
That's all a lot of the
right. Yeah, you've got the digestive system is complete.
Yep.
That's all you need.
Stefan, hi. How are you?
Hello. I'm doing all right.
Where should I get new shoes?
That's a strange question, Hank. I have really wide feet, so I have to order large shoes.
Oh. Do you have a smashed pinky toe? My pinky toe is all squished.
I have a squishy pinky toe. My pinky toe nail is like a disaster.
Oh, yeah.
It's like a joke. I look at it like, why are you here?
Yeah.
What is your purpose?
I don't know how to trim it, so I just grab it and pull it off.
Yeah, and then you're like, did I get all of it? I don't know.
This is all me, too.
Ceri is horrified.
The rest of us are like, yes, exactly.
You just pull off your toenail and expect me to accept that as yes, this is a grooming thing that I do. Remove big chunks of my body.
It doesn't feel like anything. It just comes out.
Yeah, it just
comes off.
Barely hanging on.
Yeah, barely hanging on. Is that your tagline, Stefan?
Yeah, sure.
I am Hank. I'm really excited to tangent today. I don't have to do anything except host. I can't win, but I didn't have to prepare any science facts.
Yeah, you
which
can't
seems
lose.
like a great deal to me.
You can't lose either.
I can't lose. That's my tagline. Hank Green, can't lose. All right. Every week here on Tangents, we get together. We try to one-up, amaze, and delight each other with science facts. We're playing for glory, and we're also playing for Hank Bucks. We tend to go on tangents sometimes. If we deem a tangent unworthy, we will take away one of your Hank Bucks, and you can go negative. Now, as always, we are introducing this week's topic with the traditional science poem, this week from Sam.
You may know mutants as people who shoot optic blasts or can read people's minds or can run really fast. Mm-hmm. But my friend, you are wrong. The reality is mutations are everywhere. I'm telling you, this. The reason your cat has a cute squishy face is because of slow DNA changes to the whole feline race. It's why spiders spin webs and newts have cold blood. It's why sloths do whatever it is that sloths does. All these...
things you could have just changed to find a better rhyme, but you didn't.
Like what?
I don't know anything. The newts have cold blood. Many animals have many traits. You could why sloths have the fuzz.
This is an unprecedented interruption of the science time.
It's art.
All these changes in time slowly add up and become every living thing ever, like every single one. But while that is profound and amazing, I will have to admit, I still wish I could fly or shoot claws out of my mitts.
So, Ceri, our topic is mutation. What is that?
It's genetics. So
Yeah.
genetic mutations are when DNA gets changed permanently in some way. And DNA is important because it codes for RNA, which codes for proteins, which make up all the functions that all your cells need to survive.
To do the things.
To do the things that
Made out
make
of
you
proteins.
human. Yeah. And DNA codes for all that. That's why mutations can change the way that things look or act or the way their cells act, because a small change in DNA can cause a change in a protein, which changes a whole system.
If it's on purpose, though, is that a mutation? Like if I intentionally change a genome, is that a mutation?
So you can intentionally introduce what's known as a mutagen, which is a
Okay.
compound or something like radiation that can mess with DNA and cause a random change. But I think mutations are inherently random changes to DNA. And
Right.
if you insert a gene to a specific spot that you mark and like use genetic engineering techniques, that's genetic engineering. It's not mutation.
Mutation has to be random.
I think so.
Yeah.
Yeah.
Okay.
Wow. I didn't know that.
Yeah. And it can either be like one DNA base. So I don't know, you have ATGC, and if you swap one letter out for another, that's like a point mutation. And that happens all the time. Where our cells are constantly mutating.
Yeah.
We have a lot of point mutations going on all the time. Sometimes those affect the way a protein forms, and sometimes they do nothing because it's already in a non-coding region of DNA, so we don't really care. Or it can be a whole chunk of a chromosome that gets swapped out or mixed around, or
Right.
like part of a gene. And when bigger things happen, those are usually when bigger phenotypic changes happen, or things like systems break
Right.
because a gene breaks, and you
It's
needed
like a
that to
whole
see.
hunk of stuff either got taken out or got shoved in, and the protein is like, I'm going to be different now.
Yeah, or like entire genes could be copied multiple times, and sometimes that adds a lot of function to it. Sometimes that does nothing, and you just have a bunch of copies of the same gene.
And sometimes, this is interesting, you'll have the same, like a slightly different copy of the same gene, and that protein will be very similar, but somewhat different, and that will allow for example, animals that can exist at multiple different temperatures often have multiple copies that code for a different protein that's basically the same, but works better, but like actually works at a high temperature, and one that works at a low temperature.
That's cool.
All right, it is now time for... One of our panelists has prepared 3 science facts for our education and enjoyment, but only one of those is real, and the rest of us have to guess which one is the real one, and if we get it right, we get a Hank Buck. If we don't, then this week, Stefan will get that Hank Buck. Stefan, do you have 3 mutation facts for me?
I sure do.
Can you tell me what they are?
Yes, I can. Fact number 1. Several populations of humans who have lived at extreme elevations for thousands of years have mutations that help them survive there, in places where there can be as much as 40% less oxygen than at sea level. But if you go below sea level, oxygen is also much less available to us. Bajau people in Southeast Asia who spend much of their time fishing underwater have a similar mutation, which gives them an increased level of hemoglobin in their blood, allowing them to store more oxygen and dive for over 3 minutes while holding their breath. Fact number
This
1.
is a good fake fact. I like that fake. That's a good one.
Number 2. There are people with a mutation that has been linked to low bone density, but a different mutation of the same gene can also cause extremely high bone density, making their bones seemingly unbreakable. One downside to this mutation, though, is that these people can't go swimming because their unusually dense bones cause them to sink.
Okay, great fake fact. I like that one. That's very good.
Fact number 3. 38 members of a single family living in a small town in Italy have been found to have a genetic mutation that can be traced back to a single family member that was born in 1780. The mutation has been linked to significantly lower rates of lung cancer, so all of them are heavy smokers. And some of them being in their 80s and 90s, none of them have gotten lung cancer yet.
So I feel like I've heard about a weird Italian family for sure. I don't remember what it was that was weird about them.
I feel like there's so many stories of this one mutation is just this family, and so they
Yeah.
do this thing. I don't know, like I think I read something about HIV resistance in certain families or like groups of people that
Yeah.
they were resistant to the plague, I think.
Same people were resistant to the plague and to HIV?
Yeah, and I don't know how those things are connected because one's a virus, one's a bacteria infection, but...
Okay, so I'm going to ask you about this Italian family and their lung cancer.
Okay.
Why? Like, just because I'm not going to get lung cancer doesn't mean I'm going to smoke.
Yeah, I like that you presented it like they
like,
smoke
of
because
course.
of
Now that you know you can't, like,
I mean,
but also it's obnoxious.
I mean, maybe they just think like, oh, we seem to live a long time regardless of what we do, so we might as well do all the bad things
that we enjoy.
I'm just speculating.
Do they also just like eat like sticks of butter?
Yeah.
Let me go ask them real quick. I'll shoot them an email.
I would do that.
Eat butter,
sticks of butter?
or
If I knew
email
that
them?
it was fine, I would eat.
I feel like if I knew that I wasn't going to get lung cancer from smoking cigarettes, I would still just vape. I don't vape. I actually vape.
Wow, dude, I didn't know you're that cool.
Everybody looked at me really hard. There was a lot of silence there.
So what were the other ones?
So also, to do the things, we had humans in Southeast Asia, number one, who can dive longer because they have more hemoglobin in their blood.
I feel like I've seen like a documentary on.
Yeah, I've definitely seen
feel
these
like
divey people.
A
and
pretty
they-
video of some dude
Yeah.
walking along on the bottom.
Yeah,
of the,
people who
you
are
know.
good at diving, and there's a reason they're good at diving.
Right.
I
Yeah,
can't
that's
remember what
definitely
it
a
is.
thing. We also have high bone density people who have nigh unbreakable. What was the word you used?
Seemingly unbreakable.
Seemingly unbreakable bones.
What does
Okay, I
that
have
mean?
point of order.
Deliberately vague.
I have
but
never
they
broken
sink.
a bone,
Oh,
and I am terrible at floating in water.
can you swim at all?
I can swim quite well, but I
Okay.
have to swim. I can't float. All the like floating tests, I can't do it. I always sink.
So and you sink.
And I sink. So this is real. I don't know.
I have decided I am an Italian family of 38 people who like to smoke cigarettes.
Oh,
Wow.
that's nice.
You are? Just
I
you?
didn't-
I am.
Wow.
know that about you.
I'm going to go with bones because I have broken a bone and I float really easily. So, opposite of Trace, using these two data points, that's my guess.
I feel like that's all you need in science is 2 data points.
Yeah.
Yeah, totally. totally.
I got a good gut twinge about the diving people. It sounds familiar.
Mm-hmm.
Sounds real. Think I heard it before.
Yeah, same. I
That's
feel
it.
like that documentary video just keeps playing in my head. It's like 2 seconds long.
Yeah,
Like a GIF.
and
or
it's
a
like
GIF
the
if you're a GIF
sun's
person. I
filtering
don't
through
know,
the
Josh.
ocean and someone's swimming to the bottom.
Definitely.
And they're
so I'm going to
saying-
go
Yeah.
Yeah.
divers.
Stefan.
The real fact was the bones.
The bone! I
The dense
believe the
bones.
bones. That seems extra fake.
No, it was extra real. Just a normal amount of
Can
real.
they? So you said they can't swim.
Yeah, so they all seem to be related,
Uh-huh.
and they discovered it because the one guy got into a car crash and like didn't break any bones. I assume it was a bad enough car crash that they would have expected him to break bones.
Mhm.
And then they sent him to a bone doctor, and he was like,
Bone doctor. What
the
happened?
bone
to
doctor was like, "Hey, you've got bones that are 8 times denser than normal. That's
8
weird."
times denser than normal.
Yeah.
That is
What?
a lot.
So they ended up tracking down all these people, and there's like, I don't know how many, but there's a bunch of them that seem to be related, have this same mutation, and they all have super dense bones. None of them have broken a bone
Wow.
in their lives. And one of them could not get a hip replacement because they weren't able to screw the prosthetic into that person's hip.
Wow.
What?
They-
Yeah, it's very weird.
That is
and,
cool.
And the guy who got into a car accident said that he could not swim and always sunk.
I mean, that's really dense. 8 times. That's a lot. Were the other? So, but tell me
So-
about my Italian family.
So
They're
this
real,
was-
right?
Their Italian families are real.
Fact.
It's like based on an article from 1994 in the LA Times that was about an Italian family of 38 people who had a mutation that made them resistant to heart disease.
Okay.
And they were all smokers or almost all smokers for some reason, but they were like eating.
Yeah,
like,
smoking does cause heart disease.
yeah, that's true. But also lung cancer.
Yeah, that's more of the
That
risk.
seems- yeah. So it doesn't really make sense. But they had like terrible diets and were like taking advantage of this
They're
mutation.
living life to the
Living
fullest.
it
So it was real. It was real, at least according to a 1994 article in the LA Times.
Yeah, which I didn't verify. So who knows? But I was
big
making
fact.
it up. So it's all good. Stuff I said about the people living in high elevations is true. At least for the people who live in the Andes, they have more hemoglobin. People who live on like the Tibetan plateau and other places don't. It's like a different way that they cope with high elevation. But the Bajau people in Southeast Asia the way that they cope with diving for a long period of time is by having larger spleens, up to 50% larger. So it turns out that for humans, just in general, you have in your spleen like a bunch of extra blood. And then in response to diving without equipment, one of the things your body does is squeeze all that blood out into the circulatory system so that you can, I think part of it is maintaining the pressure inside like your lungs. And then part of it is just so that you have more oxygen.
Yeah, because it specifically stores oxygenated red blood cells, right? So it's like you have that extra store that doesn't need to flow through your whole body. It's just there, ready, squish out into your blood system.
Yeah. Little
I
blood
honestly didn't know what the spleen was for.
I think it does
I
other
thought it was
stuff, too.
organ that cartoons made up.
Yeah.
I swear to God, because they're like,
It's a
and
real
I'm like,
thing.
that
Apparently.
sounds like a funny thing that they made up.
Yeah. Next up, we've got the fact off, but first, a word from our sponsors. Welcome back, everybody. We got our Hank Buck totals for you. Sarah, you have 1 Hank Buck. Trace, you got nothing.
Oh, man.
Yeah, I know. Me too. It's okay. Stefan, you got 3.
Cleaning it up, raking it in, hit me with it.
Sam's got 1. Do you have anything to say about that?
That's about as many as I normally have.
All right. So I'm very excited for Stefan to be so far in the lead.
Oh, me too.
Oh, man. Now it is time for our Fact Off, where 2 of our panelists have brought science facts to present to the others in an attempt to blow their minds. The 3 of us have a Hank Buck that we can award to our favorite fact, and to decide who goes first among our Science Couch participants here, Ceri and Trace, we're going to go back and forth naming X-Men, and this is going to be judged by Sam.
I'm so
I know 'em all.
stressed out. My heart is beating so fast.
Trace, you go first because I think you have an advantage.
I'm going to go with Cyclops.
Sure.
He's
Great.
got a thing.
Yep.
Maybe the
That's one.
easiest one.
That's a good
Yeah.
one.
Well,
That's
the
fine.
second easiest one.
Oh, gosh.
Jean Grey.
Is one. yes.
Perfect.
Beast.
Great.
Good.
Quicksilver.
Perfect.
Wow,
Yes, he's
yeah.
a
Go a
mutant.
little more obscure
Very
there.
obscure.
Yeah.
I don't know what's obscure or not.
I'm going to go with Professor X.
That is
Classic.
one.
Great.
Yeah.
North Star.
Oh my gosh! Wait,
North
is North Star a mutant?
I've never
Star.
heard
I
of
have to
this.
Google that.
I have to too. North Star's in Alpha Flight, but I think he's a mutant. I think Alpha Flight's all mutants.
I'm going to go with Angel.
Yeah, mutant. Perfect.
He's got wings.
From Buffy?
But that's all he does.
Yeah, Angel from Buffy.
Yeah,
he's a vampire
the
mutant.
ensouled demon vampire.
Flame Boy.
Flame Boy?
No.
Human Torch.
You remembered. You can't just say Flame Boy and then come back after we say no. Okay, Trace, you go first.
So, my science fact today is about your eyeballs. So, in your eyeballs, you have rods and cones, and they enable you to see in the dark and also to see color. The rods do low light vision, and the cones do color and also spatial acuity, right? So, we have 3 different types of cones, though. Mostly people just think of them as either one or the other, but there are 3 different types of cones, one for each of the major color groups that we can see. They're each sensitive to blue, green, or red. Now, those cones are coded by the X chromosome. That's where they live. So that's why sometimes men specifically will be red-green colorblind. But only red and green are coded on the X chromosome by the OPN1 family of genes.
Huh.
And so the long is the red, the medium is greenish yellow, and then the short is a blue. The name for that is trichromacy. It means 3 colors. And then that's why it's rare for people with 2 X chromosomes to have red-green color blindness, where people with XY have the regular sight sometimes and sometimes have a dichromacy or 2-color sight. So that brings me to the blue receptor, which is coded on chromosome 7. And each of the cone gives you 100 shades. So 100 cubed, you get about 1 million colors if you're a trichromat. But because blue is separate, you could also have a mutation on the X chromosome of your red-green chromosome, giving you a shift not away from red-green, but into a different spectrum altogether.
Oh.
So then you can have 4 cones. So that would be called tetrachromacy, and the first evidence of it was in 1948.
This is a thing that people can have?
Humans,
This is
women
wild.
specifically,
Yeah.
can have tetrachromacy. Oh, it's been also seen in a lot of different animal species.
Sure.
You know, fish and like mantis shrimp have, I think, 5 or something. It's crazy. I don't know. But 4-coned women do exist. These different groups of people, they think roughly 12% of women might be tetrachromats and not
the
know
heck?
it, which is so
How
cool.
How do you
Far
know?
into that range can they see?
So it depends on your mutation, but they think essentially if you imagine 100 shades with 4 cones, you could see 100 million colors. So you can see potentially millions more shades than an average person. And the problem is, which I think is just amazing, we have trouble finding them because they don't know that they're
Right.
seeing more color. That's just how they lived their life. So they said we would never be able to make a satisfactory match because the participant would be able to sense color gradations beyond those available on the test, which is great. And I thought to myself, a dude wrote that.
Yeah.
Because if a tetrachromat scientist created a tetrachromat test, they could potentially see the
Right.
same
Yeah.
thing.
So,
that would
so
be
you
really
have to
cool.
like find out that you're a tetrachromat somehow, either
Yes.
through genetic testing or by being like, you can't tell the difference between those things.
Right. Yeah. And so then there's this woman who's British, who's an artist and has been interviewed a bunch because she's one of the tetrachromats who's like out there, I guess, one of those mutants. And she said, quote, when she looks at leaves, just like regular old leaves on trees, she sees around the edge orange, red, or purple in the shadows. And you might see a dark green for you, but I would see violet, turquoise, and blue, like a mosaic of different colors when she looks at leaves. And I'm just like, what is that?
You just crushed my dreams. I was like, maybe I am. Maybe I'm one of the 12%. I have 2 X chromosomes.
But then,
But I don't
But
see
all
leaves
leaves
like
are
that.
normal.
Yeah, leaves
Yeah.
look pretty normal to me. I'm good at the gradient tests online where you can like, let's sort these colors by a gradient.
It's neat because it's like would be impossible. Color experience is so personal.
Right.
There's no way to know what someone else sees, right?
Yeah.
So they said, well, even if you asked her to describe what she can see, it'd be like trying to describe red to someone who was born blind. Like, how do you describe that?
Any color,
or like
yeah.
what the 4th dimension looks like to us boring 3-dimensional people.
The thought, like, do we all see the same colors as the same colors? There's no way to know.
Yeah.
Thought. I feel like is one that everybody has at some point in their life, and it's just like, oh man, I gotta tell my dad about this.
So what you're saying is,
Yeah, my dad. You don't go to your dad every time you have like a cool philosophical thought.
No, he texts me his a lot though.
Ceri, do you have a fact for us?
No, I just didn't bring one today.
Oh, well,
No,
Trace
I do have a fact. I thought it would be fun. It's not funny. It was a bad joke. Cut it, Sam.
No, I can't. It's too late.
Okay, so a lot of the time when you hear about mutagens, radiation comes up. Like it's in all the superhero stories. Radioactive spider, radioactive gamma rays, or things like that. Cosmic radiation, we're worried about. DNA damage from UV light. But chemical mutagens are really fascinating too because of all the ways that they can mess with cell biology in like multiple ways besides just damaging the DNA directly. So I'd like to introduce you all to a compound called colchicine, which is a toxic chemical found in purple flowers called the autumn crocus. Specifically, it's an alkaloid, so one of those nitrogen-containing compounds. And humans, by and large, use it as a medicine. So some sources have said that it is mentioned in Egyptian medical texts to treat inflammation. And that's the main genre of things that it's useful for. Nowadays, it's in gout medication, which is a form of arthritis that involves inflamed joints, or blood vessels swelling, or things like that. And we've kind of narrowed down its mechanisms of action. The big thing that it does is mess with the formation of microtubules. And microtubules are a structure that influence cell things like shape, and they can help cells move sometimes. They help with ion channel stuff. And really importantly, they help with cell division. And so during cell division and mitosis, chromosome segregation is a big part. So you replicate all the genetic material inside a cell. And then as the cell divides, you need to divide up that genetic material so you have 2 identical cells. And if something goes wrong in that process, usually bad things happen. Because colchicine influences microtubules, it can mess up the segregation step. So without microtubules, all the chromosomes could end up, like everything will get replicated, and then all the chromosomes will end up in one of the daughter cells in mitosis, and the other one will just be empty. Like it doesn't split up.
Whoa.
And that situation is called polyploidy, where you have more than the number of sets of chromosomes that you should have. So because it affects the genetic makeup of cells, papers call this a mutagen, even though it's not a point mutation, which I thought was kind of interesting.
Yeah, it's not even a mutation to the genome. It's just there's extra genome. It's like double the amount of genome every time.
Yeah.
Okay.
And so there's mutagenic activity. And I assume that, so in animals like humans, having way too much genetic material in a cell is bad. It's very bad. Our bodies don't know what to do with it. It means cell death usually.
Yeah.
But in plants, they're just chill with it for some reason. They're just fine with having multiple copies of their genome at once and can still survive.
Good on you.
Yeah.
Way to go, plants.
Or one of the big things that we've created because of it are seedless watermelons.
That's how we got those.
As is the question you got to ask is how do you get a seedless watermelon if you got no seeds?
So what they do is they soak seeds in colchicine solution to mess up the cell division in a seed. Watermelons normally have 2 copies of chromosomes, so they're diploid like humans. And so if you dose them with colchicine, they'll end up with 4 chromosomes. So they'll be tetraploid
every
instead
time.
of diploid. Yeah. You can cross a tetraploid watermelon with a diploid watermelon and get a triploid watermelon that has 3 sets of chromosomes.
Oh, wow.
Wow.
So kind of like you create a liger or a mule or anything where the chromosome mismatches. And that watermelon is seedless because if you have an odd, like
So
a
you have
mismatched
to-
set of chromosomes, it's sterile.
It's a sterile watermelon.
You have to cross it every single time.
Yes. Which is why being like a seedless fruit farmer is really, I don't know if it's more expensive, but it's more
sure,
complicated.
Yeah.
And that's it. That's the fact. So like the reason we have seedless watermelons is because of this mutagen.
Weird.
So
Cool.
That's neat.
yeah.
Okay. So we've got polyploidy watermelon seedless colchicine doped stuff from Ceri. And from Trace, we've got tetrachromatic ladies of the world have no idea how many colors they can see.
Yeah.
I have one question though. Wait a minute. So the thing with the watermelons,
Yeah.
it's not exactly a mutation. They call it a mutation. Is that just because they don't have another thing to call it?
I think it falls under the category of manipulating genetic material in a way that would happen by chance because it is
Right.
a toxin being
okay.
introduced
Yeah.
into the cell.
Okay.
It's not like scientists controlled taking the chromosomes out and putting them in.
Right.
And I think they call it a mutagen because after this happens, there's probably crossover events that happen where the chromosomes can exchange information and
Okay.
things like that.
Okay, I'm going to give mine to Trace.
Ooh.
Maybe that.
It is very cool. I would give mine to Trace too.
I'm going to give it to Ceri because I like bathing seeds in... No, I don't do this, but I like the idea that we do this. And we have to do this.
Yeah, it's one of, Stefan's got a whole bathtub full of colchicine at home.
I like watermelon and I like seeing stuff. So I think I'm going to go with Trace.
Oh, man. I got some bucks. I went from 0 to 2 so quick.
You did. Wow. It's time now for Ask the Science Couch, where we ask listener questions to our couch of finely honed scientific minds. I'm not on the couch today, so I don't have to do anything. Sam, do you have the question for us?
I do. At McLextick asks, I've heard 17% of tardigrade DNA comes from other organisms. Do larger human-sized animals get DNA from other organisms as well?
I'm going to rephrase the question. If I put a bunch of like snakes in my mouth, can I be part snake?
I'm going to go with no. What do you
Yeah.
think?
No.
Yeah,
So unanimous
no.
vote.
Yeah.
No.
I'm going to be a snake man.
I mean, you would be a snake and a man.
So you can't just steal genes from whatever you want. You can't like walk around, eat a snake, touch a plant, and be like, I want that now. But what this question is asking, and what Hank's like rephrasing is asking, foreign DNA doesn't come from like the tardigrade coming in contact with another organism and then incorporating that into its genome somehow. It usually comes from microorganisms like bacteria or viruses that work by different mechanisms, or like bacteria or viruses that use different mechanisms to incorporate their genetic material into another organism. So with viruses, humans have a bunch of old viruses
Oh, yeah.
in our DNA, which is wild to
Yeah,
think about.
It's so cool.
Yeah, those are evolutionary holdovers a lot of the time, I think. So I don't know if they've been incorporated recently, but probably way back in evolutionary time, whatever our ancestor was infected by a virus, and viruses can't replicate without co-opting a host's genome. So they integrate some of their genome inside. And if that is in a gamete, so like a sperm or an egg cell, that gets passed on to the next generation as
Usually
part of the
it
normal.
kills the cell, but like
Yeah.
if it happens to not kill the cell and if it happens to be in a gamete, then it's like, you got new DNA, friend.
Huh.
You're the winner of some
You got
viral
so many
DNA.
nucleotides.
And so that's how viruses work. So viruses are like a separate class of thing,
Yeah.
and they can insert their genetic material. But bacteria can do this thing called horizontal gene transfer. And so if you think of a parent having a child, that's vertical gene transfer, where you're passing it down through generations. And so horizontal gene transfer is passing genetic information within a generation. If you have a bacterium that somehow came up with a mutation that lent it antibiotic resistance, so when you spray it with an antibiotic, it doesn't die, then it could hypothetically, using horizontal gene transfer mechanisms, pass that gene on to a buddy and just be like, hey, friend, here's this gene that will protect you from the antibiotic. Now we can all survive together.
Here you go, Jerry. Good luck.
So horizontal gene transfer happens and has happened in the past. And with tardigrades, they're still like a relatively small and more simpler organism. And so
There are
it seems
single-celled
more-
organisms bigger than tardigrades.
Yeah. And so it seems more likely that they would be influenced by horizontal gene transfer. But a big source of contention is whether that happens in animals like humans that are more complex.
Yeah.
Because it would mean our whole idea of evolution is wild. Like, we have tons of gut bacteria in us. Are those bacteria giving us genes and inserting genes into our somatic cells and our stomach lining?
And it's starting to be, it's like literally, who knows? Like, we actually don't know.
Yeah.
I vote yes, just because that would be awesome.
Please on the
I'm
Science
super in.
Couch.
I'm
We've
super
determined
into
it.
it.
If you want to ask the Science Couch your questions, you can follow us on Twitter at SciShow Tangents, where we will tweet out the topics from upcoming episodes every week. Thank you to Geekly Unique, Patty Masha, and everyone else who tweeted us your questions this week. And now I have for you our final Hank Buck scores. Ceri, you have 2. Trace, you have 2. Sam, you have 1. Stefan is this week's winner with 3 Hank Bucks.
I did it.
Congratulations, Stefan.
Thank you so much.
If you like this show and you want to help us out, it's really easy to do that. You can leave us a review wherever you listen. That is very helpful and lets us know what you like about the show. You can tweet out your favorite moments from the episode, which we always love to see. And finally, if you want to show your love for tangents, you can just tell people about us. Thank you for joining us. I have been Hank Green.
I've been Ceri Riley.
I've been Stefan Chin.
I've been Sam Schultz.
I've been Trace Dominguez.
You can find Trace at youtube.com/Trace Dominguez, where he makes uno dos of Trace. SciShow Tangents is a co-production of Complexly and WNYC Studios. It's created by all of us and produced by Sam Schultz and Caitlin Hoffmeister. Our sound design is by Joseph Tuna Medish. Our social media organizer is Victoria Bongiorno, and we couldn't make any of this stuff without our patrons on Patreon. Thank you so much, and remember, the mind is not a vessel to be filled, but a fire to be lighted.
But one more thing. Callipyge sheep are a type of sheep with a mutation that makes the sheep amass muscle around their butts instead of fat, resulting in the sheep with extra super big muscly butts. The first known Callipyge sheep was a ram named Solid Gold. And all of his descendants have big muscly butts also. Callipyge is the Greek word for beautiful buttocks.
Gotta have one of those.
Mhm.
Hello and welcome to SciShow Tangents, the lightly competitive knowledge showcase starring some of the geniuses that make the YouTube series SciShow happen. This week, as always, I'm joined by Stefan Chin.
Hey, yo.
What's your tagline?
Why is it a pair of pants? Think about it.
Sam Schultz has joined us as well.
Hi.
How are you finding TikTok these days?
Haven't downloaded it yet. I'm not ready for that. And I feel more and more obsolete as the days go by. And I see you talking about it. And I see Ceri talking about it, especially Ceri. Because she's never known anything I haven't known until TikTok.
Mm-hmm. How's that feel?
Really bad. It legitimately feels horrible.
I'm the youth coming for you, old man.
What's your tagline?
Rain brain.
Ceri, hello.
Hello.
Now that you know about some music, what's your favorite TikTok song?
Oh, I like the one. It's the meme where a lot of indigenous folks do the dance to it, where it goes like da 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 da.
Yeah, that one.
I like that one a lot. It like brings me such joy.
See, I hate this.
Take that, Sam. And what's your tagline?
Purples.
And I am Hank Green, and my tagline is "There's a hole in my basement connected to poop." And to get
Well, it's kind of true for everyone.
yeah, but it's a direct connection in my basement.
Uh-oh.
You got sewer gas?
Yeah, I had sewer gas.
Did you figure it out?
I did. I just shoved a shirt down the hole. So fingers crossed that'll work.
No, that's right.
Every week here on SciShow Tangents, we get together to try to one-up, amaze, and delight each other with science facts. We also will give a little bit of plumbing advice. We're playing for glory, but we're also keeping score and awarding Sam bucks from week to week. We do everything we can to stay on topic, but judging by our previous conversations, we won't be great at that. So if the rest of the team deems your tangent unworthy, we'll force you to give up one of your Sam bucks. So tangent with care, unless you're me, in which case, all bets are off, baby. I'm not winning this season. And now, as always, we introduce this week's topic with the traditional science poem, this week from Sam.
You're asking me why I glow? Well, friend, I don't rightly know, 'cause I'm just a wee insect. But I'll give it a go. Here's the thing that I suspect. 2 enzymes, they intersect in my cute little rear end, and I think I am correct that one is luciferin and the other I am daring to say is luciferase, and combined they start bearing a green flash that, with some grace, tells predators, give us space, 'cause we aren't all that yummy. A flash could also showcase that we want to get chummy with a firefly honey. So there's my best guessin', even though I'm a dummy. As to why my butt's fluorescin', my shiny ass is a blessin'. It helps me in expressin' thoughts my bug head's possessin'.
So the topic for today is things that glow, of which fireflies is just one thing. Fireflies. And there is lots of glowing in nature, which is nice, because sometimes it's dark and we need to bring the light to the darkness. Metaphor.
New York Times best-selling author Hank Green, everyone. You end a metaphor.
Yeah.
You say metaphor.
That's the last lines of the new one.
Yeah, that was a sneak peek.
Yeah.
Ceri, what is glowing?
I would define it as emitting light in some way.
I was a little torn. So does like a street lamp glow, or does that shine, or is that the same thing functionally speaking?
I think generating light, releasing photons.
I think too, if you're gonna glow, you kind of have to like make light, but like in a very sort of like comforting, motherly way.
Mm-hmm.
So like a street light can glow, but it depends on my mood. It depends on how it makes me feel.
So would an anglerfish make light in a comforting, motherly way and then just eat you?
Yeah, because I'm comforted, so I'm swimming toward the light.
Mm-hmm.
And then like if it was shining, I'd be like, my eyes.
Would you say glow sticks are motherly?
Are they soothing?
I like to look at them.
I don't usually associate them with comfort.
Yeah, that's party time glow.
Yeah, like my 8th hour of dancing at a rave is not like that just reminds me of being a little babe snuggled upon my mother as we drift off to sleep. But maybe, I don't know. How do you guys not rave? Is that just me? Am I the only raver in that room?
I've never raved.
I obviously have never raved.
Yeah, I haven't either, if we're being honest.
Oh, you haven't? I would say if anybody here, you'd be most likely to rave.
I think that's true.
I probably got close a couple times.
Well, what's glowing? We never even talked about it.
I took the broadest possible definition. I looked at fluorescence and phosphorescence and luminescence, and they're all just like emitting light in various ways, whether they absorb light at a different frequency or generate it through a chemical reaction or like store it up and then re-emit it later on in time.
I love those ones to store it up.
It comes from the Proto-Indo-European root gel, which means to shine, with derivatives that relate to bright materials and gold.
Oh.
All right, everybody. So I guess now that means it's time for which is the part of the podcast where I have brought in 3 facts, but only one of them is true. And the rest of you have to decide either by deduction or wild guess which is the true fact. And you can play along at home at twitter.com/SciShow tangents, but make sure you listen to the facts first, because it won't make any sense if you don't otherwise. And I've brought in facts about Foxfire, which is not just what my in-laws call their web browser. It's also the name of the glow of bioluminescent fungi that grow on decaying wood. And people have been noticing this glowing fungi for a long time, going all the way back to Aristotle, and I'm sure before that, but Aristotle described it as a cold fire. Over the centuries, people have found clever uses for this glowy fungus, including one of the following things that was used by Americans during the Revolutionary War. Fact number 1, Betsy Ross stitched the stars of the first American flag with threads dipped in a mixture of bioluminescent mycelium, which made it so that they would glow when in the perfect darkness of the night, which everybody thought was really dope. Or fact number 2, the compass on board the first wartime submarine, which, yes, happened during the American Revolution, was hard to see because it was dark in there, so they illuminated it with Foxfire. Or number 3, members of the Culper spy ring, which was also a real thing during the Revolutionary War, would place a small batch of Foxfire to signal that a house was a safe house, so that members would know where they could find safety, even in the middle of the night. So we have these 3 facts. Betsy Ross made the stars of the flag glow, or we used it to make the compass in the first wartime submarine glow, or it was used to mark safe houses by the Culper spy ring, a ring of intelligence agents that operated around New England.
And true or false, those are that's real. Those are real. That's a real spy ring?
Yeah, that's a real spy ring.
Okay.
There was a real flag. There was a real submarine. All those things are real.
There was a real first American flag, yeah.
Whoa.
I cannot believe that the first one would have glow-in-the-dark stars and every other one would not have glow-in-the-dark stars after.
Well,
that's too good of an idea.
Maybe it wasn't widely available. I don't know.
Yeah, maybe a pain in the butt. Also, there are definitely flags with glow-in-the-dark stars.
Yeah,
I bet you can get those on Amazon.
One should.
There's no reason not to.
The other thing to notice that this wouldn't be a forever effect. It would just be sort of a temporary thing.
I don't know anything about these mushrooms, but how long would it last? Maybe it would. Maybe you.
They
can't even
glow
pull them out
while
of the
they're alive. So the glowing is part of their metabolism.
Okay.
And then so as they die and their metabolism starts to slow down. And I don't know how long, but I know that I think it's like days.
You just missed the flag a little bit. Keep them nice and moist.
Yeah. The submarine, though, are they rowing? How does the submarine work?
I honestly couldn't tell you.
It's like the Flintstones
how it was powered.
Car. Your legs stick out the bottom.
Yeah.
And
Just on the bottom.
They got flippers on.
What's the third? Oh, the spies.
spy
That one
ring.
seems pretty reasonable, but everyone would know that it glows. Like anyone walking by will be like, "Oh, that's a weird glowy thing."
But you
So it doesn't
wouldn't
seem-
know that it was marking the spy houses. You'd just be like, "Cool fungus, dude," not knowing that you are talking to the leader of the spy ring.
Is it a common fungus?
It is apparently fairly common.
Okay. Because there's not here. Do they have glowy fungus in Montana? Or is this
I
an
don't
East
know.
Coast thing?
Well, it's also wherever Aristotle was.
Ah. Where was Aristotle? I actually don't know.
Somewhere around that,
Yeah. Where all the
that area.
smart guys were from back then.
Italy, Greece, something like that.
Would they have to cultivate the fungus? Can you tell us anything? Did they just pick a tree that had fungus in it and then bring it back to their house, or did they bring the fungus and then grow it outside their house?
They would have to gather it and then put it in a place.
Got it.
The other thing about this fungus is that it often doesn't glow particularly brightly. It's not going to be right up in your face that look at this guy's glowing door. You'd have to be paying a fair amount of attention to notice the glow.
Right. Ceri, you pick first.
No, I want to go last.
But you're so smart.
Ceri maybe knows something.
Yeah, I may or may not have suggested a fact for my fact off, and then Caitlin was like, "You're already talking about this."
I will go with the spy houses.
I will go with the submarine for similar reasons as Ceri.
Oh no!
Oh.
I'm gonna go with the submarine too.
God dang it!
I tried to do the submarine a year ago for some different episode, but I couldn't find enough to make it a whole fact off. Perfect torf, though.
Yeah. So this was potentially suggested by Benjamin Franklin, who actually apparently used Foxfire to read at night because candles were dirty. And that was good for being used in the submarine because the submarine was made out of wood, made out of tarred wood, which sounds pretty flammable to me. But also because there's only so much oxygen in there, and you don't want to be sharing it with flames.
Right.
It was like a piece of wood with the fungus still in it, so the fungus was still thriving on that wood, and then he would hold it up to the compass and the barometer to see how deep he was and which direction he was going in to try and plant bombs on the side of other ships, which never worked. And he was eventually captured. His obituary said, the pilot of this craft was, "This officer is the only man of which it can be said that he fought the enemy upon land, upon water, and under the water." And the submarine, I don't know if I told you, was called the Turtle, which is a really good name.
Yeah. But wait, how was it propelled? Did they have a steam engine in there in a wooden boat?
I couldn't tell you.
I think he had I think we had a little bicycle in there that that worked a screw or whatever.
No, it had oars that were had seals between the oars. And so he would turn the oars and then turn them and push the oars. Yeah, so there's oars sticking out of the side of the Turtle with rubber seals on the end of them so that water wouldn't leak in. I can't believe this guy survived.
I would have used up all my oxygen just trying to row the oars and getting tired.
This is so bad.
No, I'm wrong. It wasn't oars. This picture definitely shows oars sticking out of it, but this picture of a model of it shows that there are pedals that you push that turn a propeller.
Okay.
And I don't know how he was supposed to deliver a bomb with this thing.
He would go above the water, and he had a little he had a bomb with a screw on it, and he would put it into the side of the boat. But then what I read was that he went to the boat, and the boat had metal plating on it, and he couldn't do it, so then he just went home.
It's got a safety weight in the bottom so it doesn't turn upside down, which would be bad. And Foxfire, the reason it's called Foxfire is not because of the animal, but is the same root as the word false. So it's like fire, but not real. False fire.
There was a restaurant in my hometown that was called Foxfire, and everyone knew that that's where you went if you wanted to meet older ladies who had money.
Wow.
Did you go and meet an older lady?
No, but you could see it from the freeway because they had a fire on the roof of the restaurant going all the time.
You always know if you need to get out of your life, you can always go meet an old lady there.
Wow.
I would love to see Ben Franklin's reading apparatus with the Foxfire, though.
It wasn't a reading apparatus. It was just a log with some fungus on it.
That's gross.
Yeah,
that's an apparatus.
I think so. There's nothing wrong with a stick. Sticks aren't gross.
He just had a mushroomy stick next to his bed.
He was dedicated. He needed to read. He needed to stay ahead of the competition.
I guess.
I don't know who the competition is, but whoever Ben Franklin's competition is, I'm sure he was a pretty competitive guy.
Yeah.
The Betsy Ross thing was pretty much a lie. There have been people who have used Foxfire for decorative purposes before, and they didn't even necessarily, I don't even know if they had safe houses for this spy group. I just read about the spy group. It was all lies.
That's disappointing.
But you could probably figure out how to make an American flag glow if you wanted to sell that product on Amazon. But I'll leave that to the listeners. Next up, we're going to take a short break, and then it'll be time for the Fact Off. Welcome back, everybody. Sam Buck totals. I've got 1, Ceri's got 1, and Sam has 2.
And how many does Stefan have?
None.
Stefan has 0. Stefan's got nothing. All right. And now it's time for the Fact Off. 2 panelists have brought science facts to present to the others in an attempt to blow their minds. We each have a Sam Buck to award to the fact that we like the most. And to decide who goes first, I've got a trivia question for you. Milky Sea effect, or Mareal, is when large areas of seawater glow thanks to bioluminescent dinoflagellates. The largest Milky Sea area ever documented covered how many square kilometers of ocean surface? Hint, they used satellite data to figure this out.
I'm going to say 3,200 square kilometers.
Oh,
Okay.
I'm going to say 100,000.
Oh,
I don't know why. That's now that I've said it, it seems like way too big, but but let's go.
The answer is 15,400 square kilometers.
Well,
So Ceri is closer.
just barely.
I will go first. So there's a lot of philosophical and biological debate about what death looks like in organisms. It's basically a systemic breakdown and shutdown or degradation of cells. But in one species, the roundworm C. elegans, there's a surprisingly obvious marker of their passing called death fluorescence. Basically, under UV light, a compound called anthranylic acid fluoresces bright blue. It's produced by little granules throughout the worm's gut, and we're not entirely sure what the gut granules do as organelles, but there are just a lot of them, and they make this glowy thing. And so when C. elegans dies, whether it's because of a lethal injury or peaceful old age, the cell death starts at one end, usually the front, and propagates to the other over several hours. And this wave of death is started by a flood of calcium ions, triggers processes that burst cells, and explodes those little gut granules. So under UV light, there's like a wave of blue fluorescence as the worm dies across its whole body. And having this clearly visual indication of death is just like wild. You can watch videos of it, and it can help us understand how cell death might propagate in more complex organisms, for example, humans, or as a clear indication of death in lab tests that involve measuring lifespan. These scientists experimented a little bit with this and were able to knock out proteins in the worm to stop the cell death wave because of injury, not because of old age. And so that could potentially be a first step in helping us develop medicine to stop necrosis from things like injury or infection or something damaging like that. So like reducing the amount of fatal injuries because we don't set off this cascade.
So they stopped the cell death. Did the worm not die then?
Yeah, it didn't die. I don't think it died as quickly. I think it died eventually because they didn't like Frankenstein it. But and here is a video.
So I'm watching death spread throughout a worm.
Yeah, it's like kind of eerie if you're thinking about it. It's like I'm watching these worms die. It makes me think that if people were able to see this centuries ago, for some reason they shined UV light on these nematodes, this would be very like a poetic inspiration for thinking about death. It's like, we all glow blue and then go dark.
So like they die and then they start to glow?
Yes, asterisk. When do you declare death is? So their cells are still vaguely functional, but then once they all burst, then that's when they start glowing. So that's why I kept it kind of vague, because I didn't feel like becoming a philosopher for this episode of the podcast. So it's just like a wave of death generically. And you can interpret that to mean whatever you want.
It may not be death, but it's definitely glowing.
Yes.
Yeah.
And after the glowing is done, it did.
Stefan, what do you got for us?
Okay, so some species of millipedes basically look identical to one another. You just couldn't tell them apart with using your own stupid eyeballs. But you could use DNA analysis to tell them apart, but that's kind of complicated. So researchers at the Field Museum in Chicago found that certain body parts of millipedes fluoresce under UV light. Specifically, their genitals glow different colors and in different patterns that are unique to the species of millipede. These aren't really like millipede dicks. Male millipedes have a hole near their second pair of legs where the bluish sperm liquid comes out. And then their seventh pair of legs are adapted to transfer sperm. And so they
Can I just make you pause and tell you that you said bluish sperm liquid like I knew that already? Where their bluish sperm liquid comes out. You don't know where it comes from, but everybody knows that millipedes have bluish sperm liquid.
Yeah.
That's what they call it.
Clearly. So their seventh pair of legs are called gonopods, and they dip them in their sperm hole, cover them in sperm, and then they run around trying to find a mate. So those gonopods have different features that are unique to the species. So there's little spiky knobs or bristly looking things. And under UV light, the differences are much easier to see between the different species. They don't know why the millipedes evolved this, because they can't. I guess millipedes don't have very good vision. They said they don't even know if millipedes can see color, and they definitely can't see UV. But from imaging all these different gonopods, they were able to condense. They thought that in their collection they had 12 different species of millipede, but there were actually only 8. And so they were able to more accurately identify the specimens in their collection.
I feel like it comes up a lot that people are like, we don't know why they have these glowy stripes on them because the animal can't see them. But it seems like the animal just must be able to see them. And
they're lying to us.
Or they just don't know what the animal can see.
Apparently, we don't really know much about millipede sex either, because they do it in the ground. And if you take them out of the ground or put them in the lab, they don't do it. So,
oh no.
Oh. Well, and if they do it in the ground, then it's extra hard to see anything.
Yeah.
That's the hardest place to see anything of all places, I'd say.
You didn't really have me until I found out that they didn't just glow, they glow different colors. Oh, color and light is very weird, you guys.
I feel sad that we can't see more of it.
Yeah.
We can't see scorpions glowing and stuff. Seems like that would be useful to us.
We can with a little bit of help.
I know, but I want to see them without help. I want to know they're coming.
I feel like I would get a headache if I was like a bee, if I had bee eyes where everything glowed intensely. I wouldn't want to look at flowers. I like flowers now.
Wait, can bees get headaches?
All right.
I don't know. Okay, nobody knows.
I have no idea. I'm not even going to try to answer
Nobody
it.
knows.
Because someone who actually knows things about animals will come after me.
So, it is now time for us to vote on our favorite fact. Are you guys ready? 3, 2, 1.
Stefan.
Ceri.
Okay.
I just liked a lot. His presentation style was great. He was waving his arms around a lot. It was really doing it for me.
Wow.
You got to do a little
The subjectivity is just getting out of hand.
I see. Yeah, this audio podcast needs a visual component during recording.
You should have seen him. He was doing a hell of a job.
And now it's time to ask the science couch. We've got a listener question for our virtual couch of finely honed scientific minds. This is from @GreatPretending. Does putting a glow stick in the freezer actually make it, quote, last longer? And if so, why? I mean, I assume yes, because it slows down the reaction, whatever the reaction is that's producing the light.
Mm-hmm.
Because there has to be some kind of ongoing chemical reaction that is releasing that energy. And the molecules will hit each other less frequently if they are moving less quickly.
Yeah, they'll hit each other less frequently and also with less energy.
Less speed, yeah.
Yeah, in the collision, which means that there's less likely for something to happen. They got to hit hard.
Does it glow less brightly as well?
Yes.
Mm-hmm.
So if you want to save up your glow stick for tomorrow's rave, you can have a diminished glow stick for tomorrow.
Yeah. It's like
once it warms up, I guess it might start glowing good again.
Oh yeah.
Yeah, it just compared to the temperature. So while it's cold, the reaction will be going slower and it will be glowing less good. But if you heat it back up again, then it'll be glowing hot and bright for as long as the reaction progresses.
And it's nice because like then your glow sticks are all cold, so you start dancing and you're hot, and you put your cold glow sticks on your body, put them in your mouth because you
Wow.
like to. I don't
Probably
know.
shouldn't put them in your mouth. Should you? I feel like you shouldn't put them in your mouth.
That's what I want. Okay, so like the kids would crack them open and like, fling them at you. Was that really bad?
It's not
Oh,
horrible. It's not like you cyanide or anything.
Okay.
The things inside in the big part of the tube is biphenyl oxalate and some sort of dye that colors it neon. And
Mhm.
then the little like crackly sound is there's usually a small glass cylinder filled with hydrogen peroxide, which is something you might have in your medicine cabinet. It's really good at like removing blood stains.
Okay.
I realize that's like a serial killer kind of thing to say. I
Interesting.
don't know. Some people have periods and you bleed all over. So suck up that laughter.
Yeah.
But yeah, so that's the reaction that happens is biphenyl oxalate gets oxidized by hydrogen peroxide, which produces a bunch of different compounds, including one that is unstable that decomposes into carbon dioxide and releases energy, which the dye absorbs so that electrons get excited and then fall back to their less excited state and release photons.
Very
And so like
sciencey.
the chemicals that you're spraying on people are like hydrogen peroxide, biphenyl oxalate, just like minor irritants, but I don't know. Like it
So don't
won't.
take a bath.
won't
in
yeah,
it.
murder you.
it's like it's dilute hydrogen peroxide.
Yeah.
Is the crackle like little chambers that you're breaking or
Mhm.
what? Okay.
I think it's just one chamber of like one cylinder of hydrogen peroxide.
Yeah.
Okay. Okay.
And
And then
like-
you can break it a bunch of times, but you only need to break it once.
I love breaking it a bunch of times. Chewing
Yeah,
on
it comes out
I used to
faster that
chew
way.
on 'em.
Oh my!
Breaking it a bunch of times will also speed up the reaction because you'll just like have more points of contact
Mhm.
instead of like just having
Mixes
one
faster. Tips for the rave.
Hmm.
You always come to SciShow Tangents for your rave tips. If you want to ask the Science Couch your question, you can follow us on Twitter at SciShow Tangents, where we'll tweet out topics for upcoming episodes every week. Thank you to at Tangential Otter, at Rebecca4, and everybody else who tweeted us your questions for this episode. Final scores. It's a tie game between Ceri and Sam. Stefan and I come in last, sharing it with 1 point each, which means that Stefan is still in
He's
the
still
lead.
going strong.
Still in the lead.
Rats.
And Ceri and Sam are 2 and 3 points behind, respectively. And I am 9 points behind. If you like this show and you want to help us out, it's easy to do that. You can leave us a review wherever you listen. That helps us know what you like about the show, and it helps other people know that our show is good because we want more people to listen to it. I don't know exactly why. There are business reasons, there are personal reasons.
It just makes me
but
feel
we
good,
do.
too.
Yeah.
We do, however, love all of our SciShow Tangents listeners, and it's such a joy to have you be here to listen to our voices. So thank you for being those folks. We love it. Thank you so much. If you want to tweet out your favorite moment from the show, you can do that too. And finally, if you want to show your love for tangents, just tell people about us. Thank you for joining us. I've been Hank Green.
I've been Ceri Riley.
I've been Stefan Chin.
And I've been Sam Schultz.
SciShow Tangents is a co-production of Complexly and the wonderful team at WNYC Studios. It's created by all of us and produced by Caitlin Hoffmeister and Sam Schultz, who also edits a lot of these episodes along with Hiroka Matsushima. Our social media organizer is Paula Garcia Prieto. Our editorial assistant is Deboki Chakravarti. Our sound design is by Joseph Tuna Medish. And we couldn't make any of this without our patrons on Patreon. Thank you, and remember, the mind is not a vessel to be filled, but a fire to be lighted.
But one more thing.
Glowing butt fact!
Glowing butt fact. There's a lot of glowing butts. A lot of bugs have glowing butts, including glowworms, which aren't worms, but the larvae of certain types of Australian fungus gnats. They live on the ceilings of caves in what the University of Melbourne describes as chandelier-like webs that consist of several snare lines covered in sticky mucus. And to lure their food into these snare webs in the dark caves, they light up their butts and they trick nocturnal insects into flying towards them and getting stuck, just like how a moth does to a porch light, basically. And bonus butt fact, they don't poop.
What?
All their waste is, air quotes, excreted as light.
What?
You don't believe me? You don't believe the University of Melbourne, then? Bring it up with them, huh?
They gotta poop at some point. Maybe once they turn into bugs.
No, when they turn into bugs, they don't have an anus, and so they just have to live fast, die young, because they can't poop.
Okay.
It's like YOLO, but with no poop.
You only poop never.
Yopo. Welcome to SciShow Tangents, the lightly competitive knowledge showcase. I am your host, Hank Green, and today joining me, as always, is our science expert, Ceri Riley.
Hello. Expert is a very generous term.
And also our resident everyman, Sam Schultz.
Good day, mate.
Oh, gosh.
With the reboot of the show, I'm going to adopt a new personality and accent.
Like every episode, or like from now on, you're going to be Australian?
I don't know. Whatever happens, happens.
Well, you've already lost it. Finally, our special guest for the week, SciShow Tangents editorial assistant, Deboki Chakravarti. Hello, Deboki.
Hello.
Deboki helps make all kinds of things, hosts Crash Course Organic Chemistry, also helps me answer the sciencey questions on Dear Hank and John, sometimes on my TikTok. But more than any of that is always here helping us at Tangents, make sure we get stuff right, and answer questions when we're confused. So, Deboki, I appreciate you for coming out. And all three of you, I want to ask you a question because of the topic for today's episode. If you could know when you were going to die, would you sign that contract? Somebody comes up, like a man, he looks very mysterious, he's glowing, and he says, hello, you have to say yes or no right now. Do you want to know when you're going to die? You can't do anything to prevent it. You can't take any, you can live your life differently, but you're going to die on this day, and now you know.
You don't know how it's going to happen?
You don't know how it's going to happen.
Ugh.
Okay. Is the man telling the truth?
The man is telling the truth.
You guys are asking a lot of questions for me. I'm just like, no. I have no interest in knowing.
Yes, I would do it.
I think I would do it too. I don't think it would change anything. Well, I guess that's hard to say because I won't know, but...
Yeah. I think the real gamble is if you're going to die of natural causes or not. And that would be the thing that you'd find out that would be like, oh, shoot.
Yeah, if it's 10 years from now, you're like, oh, that's not...
Yeah.
So, Deboki, tell me what you think of my reasoning here. I feel like the worst part for me about the fact that I'm going to die is that it could happen at any time.
Yes.
I don't mind the idea of dying. That's good enough for everybody else so far. I should be okay with it. But I want to be able to plan. I feel like that's a big life event to have no control over.
Yeah, I don't know that knowing when you're going to die is really going to give you the sense of control that any of us are seeking from any knowledge of death. It's going to feel like control, but at the end of the day, the idea of knowing when I'm going to die, like you guys were saying earlier, it wouldn't change anything. I know for me, it would change everything.
Oh, it would totally change.
I could not live comfortably and just the countdown. It would be too much for me to handle.
You do already have a piece of knowledge that is similar, which is that you will die.
Yeah.
Not proven, but true.
I am so sure that I would learn and then forget so fast. I'd be really productive for 2 weeks, and then I would just forget and be like, oh, well.
Yeah, and then somebody asks you, and they're like, you found out from the guy who tells you when you die, right? He came to you, and you're like, yeah, but I spaced it. It was sometime later. It wasn't around now.
Yeah.
So I just spaced it.
Yeah.
I feel like for me, and maybe this says a lot about me in ways that I don't know, it would be like any other sort of important date of I'm graduating for college in 4 years, counting down to that. And then it's giving me another milestone to look forward to, as opposed, I guess not look forward to, but just to count down to, because I'm out of those. I'm out of formal schooling, which gave me plenty of nice milestones of I can be done with this.
Ceri Riley just wants another deadline.
That's the thing I was thinking about is like also in terms of planning, like knowing that like, okay, this is the date that I'm planning for. I mean, I know myself and I know that either way, I'm going to wait for the last moment. So I might as well not know what that last moment is.
Well,
like
that's a great point. You are winning me over a little bit. That the reality is that the worst feeling is knowing that you procrastinated up to the point of your actual death.
Yes.
It was like for me, I can be like, well, I didn't make it to Ireland, but I didn't know.
Yeah.
I didn't know if I was gonna die or live.
Yep.
But if I die and like I knew it and I was like, man, like it was on my bucket list and I just didn't fucking do it. Like I knew when I had to get it done by and I didn't do it.
Yeah.
All right, everybody. Thank you for answering my dumb question. Every week here on SciShow Tangents, we get together to try to stump and one-up and amaze each other with facts about our world. And we try to stay on topic, but the name of the show is Tangents, so we're not great at it. Our panelists here are playing for glory, but you're also playing for Hank Bucks, which I will be awarding as we play. And at the end of the episode, one of you will be crowned the winner. And now, as always, I will introduce this week's topic with the traditional science poem. There are a lot of people who are working very hard so that each of us can spend less time in a graveyard. They stay up late at night because they want to cure diseases, from the most severe to just the ones that give you little sneezes. And if they just keep working, work better, and work fast, might there be some limit where a barrier's surpassed? Our lifespans do get longer with every passing year, and sometimes those leaps happen faster as new breakthroughs appear. So what if lifespan increased by 1 year for every year? That's what people mean when they say the singularity is near. Download our consciousness into computers for all time, create a cryogenic bath that keeps us in our prime, rejuvenate our bodies with techniques we can't foresee, and soon we'll find we are on our way to immortality. That is until the sun explodes or we give up on our endeavor. There isn't much that makes me think that anything's forever. Our topic for the day is immortality. One thing that I know and would like to say up front to all the people listening is that you too will die and nothing lasts forever. So let's find what we value and move forward from there. Ceri, what is immortality?
Well, on that cheery note, mortality is what we're all subject to, the fact that we're going to die someday, and immortality is not that. So it is just not dying.
Are there ways that this happens? Because I hear about them, jellyfish and stuff.
Yeah, so biological immortality is usually defined in terms of senescence, which is this word that means biological aging and is the gradual deterioration of the function of biological organisms over time, whether that's like DNA stability or other genomic instability. This is where telomeres come in, and like after you've copied your DNA a certain amount of times, these like bits at the end get cut off. But there's also just things like getting worse at transferring nutrients and your cells getting old and holding their liquids and their things less good.
You actually do get less good at holding your liquids.
Yeah.
That's a real thing.
And so there are organisms out there that have negligible senescence. That hydrozoan that Hank is mentioning is one of them, where we can't see the biochemical effects of aging in ways that are recognizable to us. So like they have stem cell populations that don't die off, or they can revert back to a less mature form and then grow older again and then revert back and then grow more mature again without losing that biological function.
In order to live forever, you also have to not get killed. So like living forever, like biological immortality is the idea that like you could, but you're probably not going to because something's going to happen. Like the real problem with immortality is that like you don't get the option of natural death anymore. You will die in an accident. Like if the singularity comes, everyone who dies will die an unnatural death. You'll just get hit by a car eventually.
In my definition of immortality, at least, it's like you can stop the aging, like the natural aging of your cells, but you can still get something like cancer as a disease.
Right.
So like that as a natural cause of death, but like disease that racks your body is different than delaying the effects of aging.
I do very much want to delay the effects of aging. Like I don't hold out any hope for immortality, but I would love to be a sprightly 70-year-old. It's hard to get used to this idea, but I may be a little too old to really benefit from the effects of all the anti-aging therapies that are on the horizon, but the horizon is still pretty far away.
You got to clear the way for the kids to take over eventually. That's the thing I worry about the most. Just
That's true.
Yeah.
got to
That's
get some
true.
new viewpoints in there.
Thanks for throwing a little bit of social science into it, Sam.
Yeah.
The word immortality, I guess its root is mort, which all I know is that means death, but I guess it's named after like the god of death whose name is Mort.
Oh, I certainly don't know that. There's the Proto-Indo-European root mer, that means to die or like rub away or harm. I don't know, all kinds of bad things. But what was interesting to me is it's also the root of mortgage.
Oh, wow.
Which means dead pledge. So called because the deal dies when the debt is paid or when the payment fails, presumably because the person is deceased.
All right, it is time for our first game of the day. It's a game that I have developed with a little bit of help from Deboki, but I didn't let Deboki know the answers. So this game that we're going to play is called What Lasts Longer? And I'm going to go round robin and I'm going to ask you each. I'm going to give you a pair of two things and you're going to tell me which of those things lasts longer. Is everybody ready for the thing?
Seems straightforward enough. Yeah.
All right. We're going to start out with Deboki. Deboki, you're going to answer me this question. What lasts longer, the average molecule of oxygen-20, the isotope of oxygen that has 4 extra neutrons, or the average bowel movement?
A human bowel movement?
The average human bowel movement, yes.
Important clarification. Yeah.
Okay, I'm gonna guess the bowel movement.
Deboki, most atoms around us will live for as long as the universe, but oxygen-20 exists for only 12 seconds on average before it decays into other things, and that is less time than it generally takes to poop.
Mm-hmm.
I think that you could probably get one out in less than 12 seconds, but on average, it's more like, I think I saw a generalized minimum of 16 seconds, and I was like, who did this piece?
Yeah, I want to know what the start time and end time is.
Yeah. If it's 16 seconds, it's not from when your butt hits the seat.
I mean, unless you got to the seat late.
All right, we got to keep moving. Sam, this next question is for you.
Oh,
An
boy.
adult female mayfly
Okay.
or the average YouTube video, which lasts longer?
Oh, shoot. I bet there's so many second-long YouTube videos. Or is there a minimum upload? I'll just go with the mayfly lasts longer.
Than a YouTube video.
Yeah.
People usually talk often about how mayflies don't last long. They're famously short-lived. And I sometimes hear that they survive for 1 day, but there is a species of adult female mayfly that exists for 5 minutes
Oh my
after
goodness.
hatching into its adult phase. During that time, they have to mate and lay eggs. That is half the length of the average YouTube video now,
Wow.
which are over 10 minutes long.
That is very surprising.
Ceri, answer me this. What lasts longer, the Titanic or the African pygmy mouse?
The Titanic from like point of construction to point of
From
iceberg.
the moment it hit the water to the moment it was beneath the water.
Okay. I'm going to say the mouse.
Lasts longer, and you are correct. The Titanic lasted for 308 days after it hit the water, and the African pygmy mouse is a very short-lived mammal, and they live for around 2 years. Deboki, it's back around to you. A pigeon or a dandelion, which lasts longer?
I feel like dandelions could last a while. I think plants are hardy. I'm going dandelion.
Dandelion, on average, can live for 12 years. A pigeon is actually a very short-lived bird at just 6 years.
Oh.
Sam, we're back around. A cat, or a termite?
What the hell? This seems like a double trick question. I'm going to go with a cat.
No, Sam, no. The average termite, now an individual termite lives less time than a cat, but a termite queen, can live for over 50 years, and we are not sure.
What?
We think that they might live up to 100 years.
Oh no, that's
What?
horrifying.
to be by far the longest lived insect. It's a bit of a mystery. And other insects that live that long tend to be dormant for a lot of their lives, whereas the termite queen is just doing its business the whole time. So termite queens might outlive people.
Whoa.
Ceri, a chameleon or a peacock, which lasts longer?
I'm going to say a chameleon just because.
Now, a lot of these ones that I pitched to you, they are actually quite close. This one is not at all. You are wrong. Chameleons live 1 year. Peacocks live 20. Deboki, an ostrich or Harry Potter's parents? Which lasts longer?
They were like 30 something, right? Let's go ostrich.
You are correct, but you were wrong about how old Lily and James Potter were. They were 21 years old when they were murdered by Voldemort. Ostriches can live up to 75 years. Sam, the Gutenberg Bible or a Tuatara? Which lasts longer?
Oh, God. The Gutenberg Bible.
You are correct, Sam.
Okay,
You did it. Tuatara, unlike chameleons, can live over 100 years.
Ah.
So you think they look like lizards, but they are not closely related. And Tuatara, for some reason, live a very long time. The oldest one is 120 years old, and he looks very cute still. I saw a picture of him, whereas the Gutenberg Bible is much older than that. But it's a science podcast, so I'm not even going to tell you how old.
Okay.
Ceri, the oldest lichen or the oldest coral, which is older?
Oh, I'm going to say the oldest coral just because marine things have figured it out earlier than land things.
I hear you on that, but
Oh,
you
no.
are wrong.
Oh, no.
The oldest coral that we have found is a deep sea coral. It's over 4,000 years old, whereas the oldest lichen we have found is also in a cold climate. So this is a sneaky thing that when it's colder, you can last longer. Is over 8,000 years old.
Very
Wow.
rude.
Old,
Took
old
all
boy.
the adaptations from the ocean creatures and then just went into the cold land.
Deboki, a tortoise or a clam, which is older? Which lasts longer? I messed up the game.
I'm going to go with a clam.
A clam is correct. There's a clam named Ming. Ming was collected in 2006 off the coast of Iceland in an ocean dredging experiment where they were doing some science to figure out climate change stuff. Brought Ming up, counted the rings, because you could do that on clams. 506 years old.
Oh, whoa.
Wow.
Oldest individual animal that we know of is this clam.
Did they put it back?
No, it was dead by the
Oh.
time they got to it.
Okay.
Oh.
But we imagine that there are others like that out there. And does it hurt? Does it hurt a little bit? It does. That's correct. Sam, the oldest shark or the oldest whale?
Oh, the oldest shark.
You are correct. The bowhead whale lives over 200 years. That's the oldest whale. The Greenland shark famously lives over 400 years. How does it do that? As far as we can tell, by doing basically nothing ever.
Yeah, I love those guys.
And finally, Ceri, what lasts longer, a sun-sized star or an Earth-sized planet?
Oh, no. A sun-sized star? Yes. I don't know anything about space. You chose a good one for me.
An Earth-sized planet can last for as long as it wants, as long as it doesn't
Oh,
get eaten by a sun-sized star. But stars have a lifespan, whereas planets are just rocks.
I knew that one.
I overthought it. I don't know.
Yeah, you did.
I panicked.
All right, final score. Ceri, did you really only get one right?
Wow.
I'm really bad at this game.
Wow, Ceri only got one right, 1 point. Sam had 2. Deboki, all 4 correct.
Yes.
What the heck?
Reigning champ of this one game.
Well, I guess that's why you're in charge of making sure we get stuff right. Next, we're going to take a short break, and then it will be time for the Fact Off. Welcome back, everybody. It's time for the Fact Off. All 3 of our panelists have brought a science fact to present to me in an attempt to blow my mind. And after they have presented their facts, I will judge them and award Hank Bucks in any way I see fit. And to decide who goes first, I have a trivia question. As of the recording of this podcast, the oldest verified living person in the world is Kane Tanaka. How old is she?
I'm going to go first if that's okay. I'm going to
Yeah,
guess
do it.
110.
I'll go 115.
Ooh, big number. Ceri.
I'm going to go 114.
Oh, boxing Sam in a little bit.
Yeah.
Kane Tanaka is 118 years and
Oh,
31 days old.
Wow, congratulations.
Yeah. The oldest person on record was 122. Kane Tanaka's coming for that record. Wouldn't it be wild if you were 80 years old and you're like, well, I'm on my way out, and then 30 years later you're still kicking?
Yeah.
So that means that Deboki gets to decide who goes first.
I will go first. So, In 1956, some scientists wanted to know what would happen if you shot a massive amount of gamma radiation at some canned ground meat. So they used a dosage that is 200.
I'm sorry I interrupted you with that.
I don't think Sam and I can beat that lead. Yeah.
Okay. They used a dosage that is 250 times higher than you would need to kill E. coli. So this seemed like a pretty respectable way to kill everything in the meat, except that something survived. A new bacteria, which they named Micrococcus radiodurans, though they later changed the name to Deinococcus radiodurans. The big reason why Deinococcus should not have been able to survive all of that gamma radiation is because that type of high energy radiation can break the bonds in DNA. And usually, like if you break one strand of DNA, that is bad and you need to fix it. If you break both strands of DNA, it's really, really bad and even harder to fix. And so it usually doesn't take that much damage like that to kill an organism. If you're an E. coli, you can get to like 8 to 9 of those breaks per like copy of your genome. And at that point, that is like when you will probably die. But if you are Deinococcus radiodurans, though, it takes about 275 breaks per genome copy to become lethal. So it's able to withstand about 30 times more DNA damage than E. coli. So it turns out that Deinococcus isn't any better at preventing that damage, but it is much better at surviving it because it's basically like the Wolverine of DNA repair. So while like all of our cells, they have these types of different DNA repair mechanisms, Deinococcus's enzymes and pathways are just like way better at it. It has multiple genome copies, which means it can have more than 1,000 breaks to piece together, depending on like how much damage you've inflicted on it. So it's basically this like semi-dead organism that can only revive itself by putting together 1,000+ piece puzzle that it also cannot get wrong. And again, there are a lot of DNA repair mechanisms out there, but Deinococcus radiodurans is just like operating at this whole other level. These methods likely evolved to protect the organism if ever gets like dried out or desiccated, which can also damage DNA. But it's also not like DNA is the only thing that gets damaged during desiccation or radiation. There are other molecules in the cell that are going to be damaged too. So it's still not even clear, even though we know that it has these mechanisms in place, it's not clear even like how they can initiate those mechanisms. So scientists are still studying the microbe to learn more. And of course, because we are humans, that means we've done the only thing more natural to us than shooting tons of gamma radiation at canned meat. We've shot Deinococcus into space, where it spent a year outside the ISS exposed to the sun, and it has also survived that.
I mean, panspermia is totally a thing. We're not from Earth. We came from somewhere else. Okay. I have a question. Did they leave the meat out for a while, or does like all canned meat have a bunch of Deinococcus radiodurans in it?
I do not know. So there were a lot of papers that are talking about this papers, but the original research paper itself, the methodology, I was having a hard time hunting down. So I don't know how long this meat is like lying around, but I think we're probably okay. I don't know. I hope.
Well, yeah. I mean, my guess is that Deinococcus radiodurans probably doesn't do that much damage to a person. I wouldn't want to eat E. coli, but if you're going to like, if it's like way too hard to kill every single bacteria, don't kill the ones that aren't going to hurt me. Yeah. I'm eating some right now. All right, Ceri, you are second in guessing the age of Kane Tanaka. What is your fact?
So like the hydrozoan species we briefly mentioned in the definition section, one of the weirdest natural phenomena to me is reverse development, where organisms are like, nope, conditions are not favorable right now, so I'm going to go back to being baby and grow up later. And I recently learned about...
If only the graduating class of 2020 could do that.
I hadn't heard of this before. I recently learned of a dermestid beetle named Tragoderma glabrum that is in the same family as the ones used to clean bones for taxidermy or for museums, but this beetle feeds on grains instead of scavenging flesh. And normally, this beetle's development works, the babies eat and molt into larger larvae until they eventually pupate and then become an adult. But if you deprive the larvae of food and water before they pupate, they will molt the other way, like a Russian nesting doll, and become smaller larvae instead, kept in perpetual youth. So even though time is passing, they're physiologically getting younger and reversing the genetic steps towards maturity. And this can prolong their lifespan. So normally, these beetles live from egg to death for about 8 weeks. But in a paper from 1972, 2 scientists kept larvae alive for more than 2 years through 20 to 36-ish week cycles of food deprivation and regrowth. And it could probably work for longer, but they just got bored, ? And wanted to see if the beetle larvae would pupate into adults, which they did. So this staying baby process isn't perfect immortality because they still showed some cellular signs of aging. For example, it took the larvae gradually longer to redevelop into their original size, from 9 days in the first de-age-re-age cycle to 28 days in the fourth cycle. And their fat cells had DNA composition similar to adults instead of babies, which the authors speculated could have caused those longer re-aging timelines. But as far as I can tell, this 1970s paper was the only big experiment on these beetles, but more people could be studying this. And maybe people aren't interested because humans don't molt and pupate. That's my guess. But I think someone should be studying these beetles.
There is definitely an economics comparison here. If you almost let people become adults and then you throw them in economic crisis, they will revert. That's been the last 30 years of life here on Earth.
Yeah, that explains a lot of stuff about me.
Oh God, no! I mean, none of us are grown-ups. Sam, no, don't worry. All right, very weird, very good. Sam, can you top it?
Probably not, but I'll try. So in some respects, living forever seems nice, but let's say you're a vampire and then think it out to its logical conclusion. So assuming that you weren't vampire hunted, would you live through the destruction of the Earth? Or the sun burning out? Or the heat death of the universe? Eventually, would you feel all your atoms lose coherence and drift apart? Probably, and then you'd be dead. But then what about the particles that those atoms are made of? Are they the one thing in the universe that's truly immortal, or would even they eventually die too? Well, that question, the part about the particles dying and not the vampire speculation, is what an international team of scientists set out to answer in 2015.
I love it. Great frame, Sam. One thing that we never think about is how do we make this understandable to Sam Schultz?
It's the only thing I ever think about. So now I'm going to talk about particle physics, and I'll probably be wrong, but I'm going to try. An electron is the least massive charge-carrying particle. True or false? Yeah?
True.
Cool. And we've never observed an electron decaying. And in fact, if I'm understanding my brief skim of the Wikipedia article for the standard model of particle physics correctly, a lot of the way that we understand how the universe is put together depends on the fact that electrons don't decay. For one thing, they'd have to lose their charge, which would, I think, disprove the law of conservation of energy, which seems bad. But there are non-charge carrying particles out there that are less massive, like photons and neutrinos, and I guess it's not entirely out of the question that an electron could break the laws of physics as we know them, lose its charge, and split into these smaller particles, even if we've never seen it. So the researchers went to Italy's Baraxino detector, which is basically a 300-ton tank of super pure, petroleum-based, quote, organic liquid, shielded from any outside radiation and also under a mountain to further shield it from radiation. It's lined with thousands of special cameras that can detect some subatomic particles, and I think it's basically like an aquarium, but it's full of a known quantity of particles instead of fish. So what they were looking for is there's a hypothetical idea of what it would look like if an electron decayed, and part of that idea is that the electron would let out a flash of light that would be detectable to the equipment in the detector. So they set it to detect this flash of light, and after 408 days, they didn't see any of those flashes. So there are 10 to the 23rd electrons in the tank, which I guess is a super huge number, and none of them decayed. So the researchers were able to math out that an electron's lifespan is probably more than 66,000 yada years, which is 6.6 times 10 to the 28th years, which is 5 quintillion times longer than the universe has been around.
And that's the lower limit.
Yeah. A quintillion is 18 zeros. And since the detector isn't perfect, it's probably even longer than that. That's just as far back as we can math it out right now. So the law of conservation of energy is safe for now, and we can rest easy / be terrified to know that the basic building blocks of our reality will still be floating around quintillions of years after we're gone, even ones that currently make up you, me, and vampires if we have electrons in us, which I'm only like 90% sure we do. We have electrons.
Yeah, I am 100% sure we have electrons.
Okay, cool. Phew.
Oh, God. Sam, That was really well done, man.
Thank you.
You got all that right. That
Cool.
was great. And I love you took immortality to its furthest possible extent. Yeah, I'm not going to be around. No, maybe the Earth isn't going to be around forever, but our electrons will be. And it really seems like maybe forever.
Mhm.
Though, who knows? Maybe there is a state at which eventually the universe kind of collapses into a bunch of black holes that become future Big Bangs.
Not my problem. So
Well done. Oh gosh, and now I have to choose between the three of you. I think I'm gonna give Sam. I think I'm gonna give you 4 points because the vampire thing was very good.
Mhm.
You drilled down and got a bunch of particle physics, which isn't easy, and I appreciate it. And I understood what was going on, which I don't always when we get into that. I'm gonna give Deboki 3 points because that was my second favorite thing, and I forgot what it was already.
we were ionizing cans of meat.
Oh yeah, you ionized a can of meat for me, which I always appreciate. And Ceri's coming out with 2 because it was also very good. And that means that the winner of today's episode is Deboki Chakravarti with 7 points, beating out Sam by just 1 solitary stinker. And it also means that it's time to ask the science couch. We've got some listener questions for our couch of finely honed scientific minds. This question is from @RebeccaRebec4. What is the science/reality behind cryonics? Can you really freeze something and bring it back to life? I know that you can freeze a nematode and bring it back to life, but I don't know if it gets much bigger than a nematode.
Something bigger than a nematode has been frozen and then thawed, but not a whole organism. Rabbit kidneys, for some reason, in the research that I was doing, are like the prime experimental material for preserving an organ and then thawing and trying to transplant it back into an organism. And so in 2009, a group of researchers used quick freezing techniques, and I think with chemical cryopreservants, which basically act like antifreeze. So when it freezes, the antifreeze solution doesn't crystallize the way that blood would in the kidney, which would be bad because then the ice crystals would form and that would damage cells. So they used this antifreeze concoction to preserve and then thaw a rabbit kidney, and then they put the kidney back into a rabbit, and it worked for 48 days. And at that point, they were like, looks good. And they euthanized the rabbit so they could study it. So I don't know if there have been any experiments since then that have lasted longer, but it seems like rabbit kidney transplantation is the most promising field before, I guess, in all of cryogenics, in my opinion.
I also briefly researched a company that will freeze your brain, and they say that eventually they'll figure out how to unfreeze it. They just don't
We
know.
promise.
Yeah. A lot of that, I think, in terms of like whether or not there's any like science behind cryonics, like there are companies that will do the freezing and whatnot, but the idea that they're actually like they can freeze you just fine. The idea that they're going to be able to revive you is really the crux of the issue, and that's going to be pretty tough to do. But there are cases like with surgeries where people will like the surgeons will actually cool a body down and have it go to like really cold temperatures so that there's basically no pulse, no blood pressure, like no brain activity, or at least really minimal. And that's to make the surgery easier. But like even then, like that is in a very controlled setting, and it is still really difficult. It is still pretty risky, I think.
Right.
I don't know if it's gotten safer. I think the closest we can say to like being able to thaw out a human is probably with like frozen embryos at fertility clinics, which is still
Very
remarkable,
different.
but it's still going to be a bit a ways from being able to revive a frozen full-grown human being.
Yeah, I mean, it does make me think that there's like a version of it that's just torpor, where it's like you're not like frozen with like ice crystals, but like let's put this person into a state where they could, maybe go to Mars and not have to be awake the whole time.
Yeah.
Or if you're very sick, you put somebody to sleep until they get to a place where, or a person gets to them who can take care of them or something like that.
I think something like what you're describing is also a key part of cryonics that we don't talk about as much, because usually people talk about like freezing their brain after they die, whether of an illness or of old age. And then that adds an additional problem beyond taking your brain back to life. You have to like
That you've died.
Yeah, you have to fix
what killed you also.
Yeah.
It's like if you put yourself into stasis, like if I put my 26-year-old body into stasis, then when I like unstasised, then at least my cells wouldn't have aged and I can like resume living my life. But if you're an old person and you die and then you freeze your brain, then it's like if you like rejuvenate your body, you're still old. You still like if you died of natural causes, or if you had cancer in the brain, then you still have that illness that scientists have to work on and cure.
Well, also if you've died, your body is starting to decay. Like, and so there's an ethical concern here. Like, I'm not going to freeze you if you're not dead, but if you are dead, you have already started the process of like actually returning to the soil. But there's also these things with cryonics that are potential actually useful therapies when it comes to giving rabbits new kidneys or being able to do surgery on people in situations where otherwise there would be just too much blood. All right. If you want to ask the Science Couch your question, you can follow us on Twitter at SciShow Tangents, where we'll tweet out the topics for upcoming episodes every week. Thank you to at Mama Paula Bear, at CrystalR99, and everybody else who tweeted us your questions. For this episode, Deboki, what are you working on these days? You got anything you want to plug?
Watch Journey to the Microcosmos. It's a pretty good show.
And if you like Deboki, there's also tons of episodes of Tangents that Deboki's been a guest on.
If you like this show and you want to help us out, it's easy to do that. You can leave us a review wherever you listen. That's helpful and helps us know what you like about the show. Second, you can tweet out your favorite moment from the episode. And finally, if you want to show your love for SciShow Tangents, just tell people about us. Thank you for joining us. I've been Hank Green.
I've been Ceri Riley.
I've been Sam Schultz.
And I've been Deboki Chakrabarty.
SciShow Tangents is created by all of us and produced by Caitlin Hoffmeister and Sam Schultz, who edits a lot of these episodes along with Hiroko Matsushima. Our social media organizer is Paolo Garcia Prieto. Our editorial assistant is Deboki Chakravarty. Our sound design is by Joseph Tuna Medish, and we couldn't make any of this without our patrons on Patreon. Thank you. And remember, the mind is not a vessel to be filled, but a fire to be lighted.
But one more thing. One way to immortalize poop is to let it fossilize into what's called a coprolite, a dried out piece of poo. The oldest human coprolite was found in Oregon's Paisley Caves, and thanks to radiocarbon dating, it's estimated to be about 14,000 years old.
I just love that there are some people who are like, they look down and they're in a cave and they're like, that's not a rock.
How would you ever know that in a million years?
I don't know.
I'm assuming it's one of those things where once you know, you see it everywhere.
Oh, yeah.
All you see is rocks and poop.
Poop and not poop, probably. It's a binary filter on the world.
Welcome to SciShow Tangents, the lightly competitive knowledge showcase. I'm your host, Hank Green, and joining me this week, as always, is science expert Ceri Riley.
I feel like I need an asterisk next to my science expert for this episode, too. just a little, imagine it.
And also our resident everyman, Sam Schultz.
Hello.
And we also have a special guest today, Twitch streamer and artist Ashley Roboto.
Hi, everybody.
I love having guests.
I'm very excited.
Ashley, what's the best Zelda game?
Oh my gosh. Okay. Considering this is a conversation I've had much today, I think personally,
really?
yes. My favorite Zelda game is Majora's Mask, just because I am a fan of creepier stuff, and it's one of the first games I ever played. And I love it. I feel like every time I play it, I discover something new and nuanced about it, and it's really cool. But replayability, I feel like Ocarina of Time and Wind Waker are pretty solid.
I think that I have spent the most of my Zelda time in Majora's Mask personally. So I appreciate that.
Wow.
Oh, that makes me
I feel very sophisticated.
solid.
That was college time for me. So
Yeah, I can't picture a
Oh.
version of Hank that has time to play video games.
I haven't played a video game in so long.
Oh no. I'm very sorry. Also, I won't tell you when I played Majora's Mask. I'm going to leave that age off the table then.
No, I'm used to this.
Yeah, I usually say something like I played Majora's Mask when I was little and it scared me because
Yeah.
I was too small. And so
Yeah.
the moon coming down was just like,
Frightening
this
guy.
is too
Yeah.
frightening for me.
Because
as a
Ceri hadn't
child.
been born yet. She was just an embryo playing Majora's
She
Mask.
saw through her mother's stomach the moon coming at her and got scared.
Yeah, through the belly button hole, you know?
That's how it works.
Yeah,
Science.
This is a science podcast. That's exactly how it works.
Yeah, that's why it's there. It's a peephole. I had a great conversation today with Maddie in the SciShow studio, where we both discovered that we watched Blue's Clues at the same time, but I did it as an adult because I didn't want to think about my responsibilities.
Uh-huh.
Oh,
And
very
she did it
valid.
because she was in preschool.
She
or
did it because she
less.
was a preschooler.
Oh, no.
That's awful.
This thing that you're listening to right now is SciShow Tangents. Every week we get together, we try to one-up, amaze, and delight each other with science facts, all while trying to stay on topic. Our panelists are playing for glory, but we're also playing for Hank Bucks, which I will be awarding as we play, and at the end of the episode, one of you will be crowned the winner. Now, as always, we introduce this week's topic with the traditional science poem. This week, it's going to be from me. Resistance is futile, they say with a smile, as we visit the Apple Store, walk down the aisle to see AirPods and watches we can add to our bodies. They're hundreds of dollars, but still don't feel gaudy. They connect us to others and to our own selves. They're always upon us and never on shelves. Helps you sleep, helps you wake, helps you bake a sweet cake, helps you get from your house to a good steak and shake. There's this idea that someday we might be part machine, with sensors embedded in our skin so serene, but that isn't a day that we someday aspire for, because already most of us have become cyborgs. And our topic for the today is cyborgs.
Oh.
Oh my God, we don't even get a snapping break? That was
Oh,
beautiful.
yeah, please,
we
everybody
snap
snap.
sometimes, yeah.
if it's good.
Oh.
Wow, Ceri didn't snap.
It was fine.
Wow. Sarah's been on a poem hot streak lately, so
It's
she's-
true. It's true. Look, we keep getting better at poems, and so occasionally one maybe a little bit misses the mark. I feel like that last stanza I could have worked harder on it. Well, nothing rhymes with cyborg, literally. Ceri, what's a cyborg?
Well, I think you approached the heart of the debate in
Mhm.
your poem, which is that it doesn't really have a scientific definition. So it's kind of like wherever you want to draw the line,
Mhm.
the line can exist.
Right.
But the principle of a cyborg is that it is like a shortened form of cybernetic, an organism, and is
Mhm.
used to describe something that is part biological and part technical.
Mhm.
And so for some people, it's like, well, we're riding a bicycle or we're wearing glasses, and that's like a piece of technology that is enhancing our physical form in some way.
Yep.
But some people are like, you need to have like the traditional sci-fi picture of a cyborg is someone with like a laser eye or a completely replaced chunk of their body
Right.
that not only performs the function of that chunk, but like enhances it and is seamlessly integrated with the biological parts of their flesh, as opposed to like a phone that you hold in your hand.
That was a little more broad than I have ever thought. And as soon as you said glasses, I just kind of sat here for the rest of the time thinking
I'm like,
about it. That
oh,
really makes me
yeah.
cyborg.
You're totally a cyborg.
Yeah, that's like up to you to decide as a glasses wearer. Do you want to be a cyborg?
I think it just sounds cooler.
Yeah, but also what counts as technology, right? Like language is a kind of technology. So from that perspective, humans have kind of been cyborgs from the very beginning. That's the thing that makes us so spectacularly able to survive. Maybe a little bit too able to survive if you ask some other species. And so like what is a technology and what is it integrated in? How is it integrated into us? But I really do feel like the AirPods, that makes me feel very much like I'll be across the house and then Ceri will be like, hey, Hank, you just got a text from your mom and here's what it says. And I'm like, you took the internet and put it into my brain just then. I'm not going to make Ceri tell us the etymology of the word cyborg because it seems pretty recent.
It's recent, but it's interesting. I
Okay.
was excited for this one.
All right, you
Um,
go.
It was coined in 1960 from a paper co-authored by two dudes, Manfred Kleins and Nathan Klein, unrelated, who were specifically writing about people exploring space. And they made an argument that instead of creating human-like habitats in space, so like a lot of NASA research is like seeing how human bodies are affected by space and how we can build spacecraft or like habitats on other planets to protect humans and mimic an Earth environment. They suggested that what if we solve this problem by creating cyborgs? So changing the human body to adapt to a space environment rather than changing space to adapt to humans. And that is why they came up with a term. They were like, we can't adapt fast enough to extraterrestrial conditions, so we should just become cyborgs if we want to go to space and adapt our bodies with technology instead.
I mean, it's a real win. It's a win of a term. Cyborg. It just sounds very good. I often think sometimes whether or not an idea sort of sticks in our culture really comes down to whether you come up with a good word for it. So good job, Klein and Kleins.
Yes.
That dynamic duo. And because it's a constructed word, maybe that's part of why it doesn't have a lot of good rhymes.
The only one I can think of is morgue.
Yeah, I did. I looked at morgue. I thought about morgue for a while.
Not very fun though.
No, yeah.
Fly morgue. You can make it kind of, I guess, fly the organism or fly cool. It could be a cool place where dead people hang out.
Fly more. It's not like for the dead flies, but like the hip version of fly.
Yeah, the lit one, as they say, right?
The hip and cool.
It's a fly morgue. Now that we're fully up to speed on where the word cyborg comes from and what it means, it's time for the quiz portion of our show. This week, we're playing a quiz called Secret Ingredient. So scientists have been exploring a variety of different methods to integrate plants into our potential cyborg future by using their internal biology and chemistry to create all sorts of cool things. So not a cyborg human, but a cyborg plant. This work is still very much in progress, so a lot of what they're making are just proofs of concept that might help us understand more of how we might combine plants and electronics. So for today, we're going to be playing Secret Ingredient, where I will describe a cyborg plant technology, but I'm going to leave out a key ingredient, and you will have to guess of 3 options which that key ingredient is.
Okay.
Round number 1. In 2015, researchers at the Linköping University in Sweden, I'm sure that's how it's pronounced, demonstrated the ability to add electronic functionality to a plant by assembling simple circuits that combined the rows with electronic materials. What was the secret ingredient behind their methods? Was it A, an electrode that is activated by the puncturing of thorns? B, they don't have those. Roses don't have horns. B, genetically engineered protein that amplifies voltage. Or C, a polymer electrolyte mixture that assembles inside the xylem of the plant.
Ooh.
But why?
But why what, Sam?
Why are they doing this?
I said it's a proof of concept. We don't know yet what we're going to do with it.
Okay.
Maybe we're going to make the rose do a little dance for the one that you love.
That would be nice.
You know, it's like Jurassic Park. It's like, it's about if you could, not if you should.
Yeah. Movie definitively proved that every time you should. Just keep doing. Well, I'm going to pick the xylem one.
I see.
That's a word I know.
Yeah.
Xylem. Sounds like you could fit something inside there.
It seems like the most specific to me. So I think there's the most thought there. So I'm going to go with C.
Okay.
The thorn one sounds fake. I don't know what you'd do with puncturing thorns.
Yes.
So I'm going to go with B, just for the heck of it.
You just want to add a little variety, a little spice.
Yeah.
Well, Ceri, you should not have, because the answer is C.
Oh.
So scientists incorporated electronic components into a rose by submerging the stem in a solution of a conducting polymer called P dot. The P dot goes into the xylem. It's just sucked in via capillary action, and it creates wires that run through the rose. The scientists then tested the xylem wires out by cutting bits of the stem and seeing how they performed in different circuits. So boom, rose wires. Why are we doing it? Don't ask questions.
Okay.
All right.
That
awesome.
makes sense, because that's how they make roses like fake different colors. You know, the ones in the grocery store that you see that are like bright blue. They just like put them in food coloring like that. So
one of the other things that they did is they used a different polymer, and when they run a voltage through that polymer, it changes color. So maybe that's what they're going to do. Coming to a flower shop near you, color-changing roses if you plug them in. Round number 2, everybody. Scientists at Nanyang Technological University in Singapore created a soft electrode that would allow them to monitor a plant as well as send electric signals to the plant to control its movement using a smartphone. To prove this concept would work, how did the team test out their system? Did they A, turn a Venus flytrap into a set of pliers that can grab onto little tiny wires, or B, construct a braided structure out of grapevines by controlling the movement of the vines, or C, pull a lever connected to a sunflower by controlling the plant's bending? Plants can move, and now we can make them move because we can't control ourselves.
Yeah, but why?
Yeah. Oh my gosh. See, it was super cool in theory, but now that we have like a practical application, I don't like it as much.
It's creepy.
Yeah.
So my first thought when you said moving plants was sunflowers, because I know they move pretty dramatically to follow the sun. And like even watching them in our yard, like they rotate throughout the day. That's really cool. So I'm going to just go with that one because I've seen them move, and seeing is believing. Really, I have no idea. And I was just trying to make a reasoning behind a guess. Grapevines are really flexible in the way that you can like weave them around things.
Yeah.
Like any sort of viney plant is. And Venus flytraps already have the
little mouth.
little mouth that opens and closes. And I just imagine it would be harder to pry it back open. But if you understood what electrical impulses or like what mechanism closed it, I imagine you could open it as well.
Well, yeah, it must be somewhat similar-ish to like being able to electrode like your arm,
Sure.
right? Oh no, I was kind of solid in my answer. Now I don't know anymore. But I'm just gonna guess B because it just felt right at the time when I heard it.
I don't think a Venus flytrap would be very helpful in any situation with little wires. So I'm going to also say B.
Well, Sam, it doesn't matter whether or not it's useful. You just do it because you're a scientist. Because it turns out the answer is indeed A. So many plants, it turns out, have really irregular surfaces, and that makes it hard to attach electronic devices to them. But hydrogel electrodes, similar to what are used in EEGs, are easier to attach. And to test out whether these electrodes could work at controlling a plant, scientists connected them to a Venus flytrap and then sent electrical pulses through like Bluetooth or the smartphone or something to get it to close the leaves. Now, did they get them to open them back up? I don't know. That's not in my notes. Probably not. But that's
I guess no.
Then they connected the plant to a robotic arm, and they were able to get it to pick up a little wire. And look, it's just so you can get a headline out of it. That's the whole point.
I guess so.
Yeah, fair.
And finally, round number 3. Scientists at MIT in the US connected electrodes to the stems, leaves, and ground of a potted plant, using the plant's own internal chemistry to send signals to a robot attached to the pot. The robot has a key part that allows it to respond to what the plant is feeling. What is that part? Is it A, a set of wheels that moves the pot when the plant senses light, B, a heated wall that turns on when the plant gets cold, or C, an arm that transports water to the plant when it is thirsty?
Okay, that's cute.
It's one of those, and regardless, it's awesome.
They're all cool.
Maybe robots and plants can be friends.
Yeah.
Oh my gosh, could you imagine how much it would help people that just cannot keep a plant alive? It's like, that's in the robot's hands now.
I got it.
A heated wall sounds so boring. I hope it's not that one.
I feel like C is watering, right? I think that would probably be the first place that, like, you know, if I was smarter and a scientist, I would try to make that work. I think it's C. They're all very plausible, though.
What's it do? Yeah. The first one, what was A?
A was a set of wheels that moves the pot when the plant senses light.
Maybe, wouldn't you want it to move when the plant wasn't sensing light?
Well, that's like maybe the thing is if it's not at the right level of light, then the wheels wheel it to get the right level of light.
The answer was, though, I believe, when the plant is sensing light, right?
Oh.
We're going into technicalities.
It does. It moves the pot when the plant senses light.
Suspicious. I think it's the watering one.
I think it's the watering one too, just because that feels like plants sense that really obviously.
Mm-hmm.
But the other thing that they sense really obviously is light.
Oh, no.
Oh, no.
And look, I don't know why they had the plant move when it sensed light. I guess just because they could.
Because they couldn't figure out how to do it when it didn't sense light. Maybe.
Exactly, yeah. Or if the plant isn't sensing light, it just drives itself around forever.
Like a Roomba. Yeah.
It's like just searching. I love this so much. Maybe it like moves toward the light. I don't know. I don't think it does. I think it just moves when it senses light. But there's a name for this system. It's called the Elowan, Elowan. So it sounds very elvish.
Yes.
And anytime there's a plant just driving itself around. It makes me really happy.
Someday
That's
plants
fun.
will have cars.
That is a little more scary. I like the little tiny potted little cars, though. That's cute.
Yeah.
Yeah. Someday plants will have cars and they will just drive faster and faster as long as there is sun.
Yeah.
It's like, I love this so much. And then they just crash into each other. And that's the thing that finally stops us and gets us on trains again.
Yeah, just shove a bunch of sunflowers on the Autobahn and let them go.
Exactly. Well, that means that overall, none of you did particularly well. But no one as poorly as Ceri. The scores are Ashley with 1 point, Sam with 1 point, and Ceri with 0 Hank bucks.
Oh,
Next,
no.
we're going to take a short break, and then it will be time for another game. Welcome back, everybody. It's time for Contact lenses have a long history, including one contraption proposed in 1637 by René Descartes that involved filling a tube with water and placing the tube in contact with your eye.
No, don't do it. Not in
No.
that time.
Yeah, come on, Descartes. I thought you were smart. Fortunately, scientists and doctors have come up with a lot of advances since then, and many of us have been able to correct our vision thanks to the lenses that we put directly onto our eyes, which I haven't done since high school because I just didn't like how it felt. But of course, technology continues to advance, and scientists have developed ways to integrate electronics into our contact lenses so that they can do more than just correct our vision. The following are 3 stories of cyborg contact lens advancement for medical purposes, but only one of them is real. Which one of them is it? Are you ready?
I think so. Maybe.
Fact number 1. Scientists have invented a contact lens that monitors changes in the shape of your eyeball, collecting data and sending it wirelessly to a device so that doctors can diagnose potential disease. Or is it fact number 2? Scientists have invented a contact lens that clears persistent floaters from your eyes using small vibrations that are programmed so that when worn, the contact lens will shake the floaters and clear them away.
Windshield wipers.
Yeah, basically, for your eyeballs. Eyeball wipers. Or fact number 3, scientists have invented a contact lens that helps reduce eye strain by using light-sensitive materials that trigger the muscles in your eyes to temporarily relax when you've been staring at a screen for too long.
That sounds dangerous.
Look, we've just gone over the fact that scientists are making plant cars,
Okay.
and we're all going to die.
I feel like the most useful one is the first one. It would be like monitoring that kind of nonsense,
Yeah.
which is helpful.
People are very into this, just like covering our body in sensors so that we can always know when we are experiencing or not experiencing disease. Just put
Yeah.
the whole doctor business out of business.
Well, there's like this increasing push towards personalized medicine that's also like a surveillance state that I don't know how to feel about because I'm not
Yeah.
smart enough.
Apple's
But,
like, I know how firm your eyeballs are.
Uh-huh.
Yeah, it's like, you know how many steps I took, you know my sleep patterns, and you know my eyeball firmness. And like, is that good because you can tailor my care?
Yes.
Yeah.
Maybe. Is it bad because, I don't know, you can market me things based on my eyeball firmness?
Yeah.
Yeah.
Oh my gosh. Are we going to have like a Tempur-Pedic eye firmness scale?
I've never worn contacts. I'm terrified of them.
I do not like touching my eyes.
No, hilariously enough, I've worn glasses since I was like in 6th grade. But in high school, instead of, normal contact lenses with a prescription, I wore colored contacts and then my glasses just to really make my life hell for a while. So.
Yeah. Sounds very high school.
Yeah,
Yeah.
it sounds eye infection-y.
Didn't do it for very long, so thank God.
Yeah, I stopped wearing contact lenses when I went to a Warped tour, and I lost them both in a mosh pit.
Oh, no.
And
Oh.
had to call my mom to pick me up.
Oh, you loser.
It was a loose mosh pit. There were not very many people in the audience, but there was one man who put me in a headlock and then raked his whole arm across my eyes and extracted both of my contact lenses.
Ew.
Oh, that is way too much eyeball contact for my liking.
How did you keep your eyes open through that whole process?
I don't know. It was very intense.
You were scared.
Mosh pits, man. You can't close your eyes. You never know what's going to happen.
I guess so. Wow.
Yeah.
Well,
Oh, my Lord.
Do you guys want to hear him again?
they're written down, but
I'm still
All right.
going to go with number 1.
Yeah. You've got your wireless eyeball-shaped monitoring contact lens, your vibrating floater clearing contact lens, or your light-sensitive muscle-relaxing contact lens.
do your eyeballs change shape?
Yes. Yeah, they do.
I guess when you squint, you're making them a different shape, huh?
Well, like even when you focus on things. So this is another cool part. I read an interview with the guy who made up the term cyborg, and he's like, we already kind of are because like we get muscle feedback. This is a cool tangent I wanted to work into the episode, so I'm glad you asked.
Oh, you look so
we get-
excited.
We get muscle feedback. So if you think like, oh, I want to clench my fist, you can like feel that
Uh-huh.
and like see the consequences of that. But the control of the lens in our eyes happens not quite subconsciously, but like without that kind of motor feedback. We just like think about what we want to focus on, whether it's at a distance or up close, and our eye lenses and our eye muscles adjust that for us. And like the feedback is that we see the thing that we want to see more clearly, but we don't like feel those muscles move necessarily. And I like sat after reading that and thought about it for like 10 minutes because it's just like, I don't know. I couldn't tell my lens like while I'm focused on something up close, like to focus far away or focus at a different distance. I can make it either like blurry or not,
Mhm.
but not with the precision that
Right.
my eyes and brain do
Yeah.
without me consciously thinking of it.
Yes, eyes are very much like surprisingly out of our control, and also like the information we get from them is surprisingly not what we think it is. It's a kind of illusion oftentimes.
I will still never get over just thinking about like the cones in our eyes and how different animals have so much more. It's like I have no idea what we don't see in the world, and that is an uncomfortable thing to think about.
The light-sensitive muscle relaxing thing. What if you forget that you have them in and you're driving down the street and all of a sudden you're like, "Boom." Is that what happened?
Man, I don't know.
I don't think that dramatically, probably.
Okay.
I hope not.
That sounds too dangerous. So I'm going to go with number 1 also.
Okay.
Okay, I'm gonna just try to be different, and it
No.
hasn't worked out for me
Don't do
so far.
it.
I'm gonna go with the muscle relaxing one, just because
Oh.
I don't know.
I'm going to keep you all guessing a little bit and let you know that Ceri was wrong, but was close, because scientists have been developing contact lenses with artificial irises that respond to light for people who have damaged irises. So
Cool.
if you aren't able to contract your own iris anymore, scientists are working on a thing that responds to light and contracts an artificial iris. So you are right that it is a thing, but it was not that thing that I said that it was. It was close, but not quite there. Now, did the other two people get it right? You, in fact, did.
Yeah.
Good job.
So yeah, this soft contact lens has a microprocessor in it and an antenna in it and a strain gauge, a device that measures the amount of strain the lens is experiencing. And it's used to measure intraocular pressure, which is a major risk factor for glaucoma. And you could do this when you go into the eye doctor. Sometimes they'll like shoot a puff of air at your eye, and that will give you that moment's intraocular pressure. But those measurements don't capture the variation that can happen over the course of a day. So that contact lens, which is made by a Swiss manufacturer, Sensimed, it's called the Triggerfish, which is a lovely name, and it's designed to be worn over the course of 24 hours, during which time it will take 300 strain gauge readings over 30 seconds every 5 minutes, producing 86,400 data points over 24 hours. And that is then just sent to a recorder on the patient. And the data then gets into a computer where the doctors can assess your patient's eye health so that we can know for sure how your glaucoma is doing.
Okay. So do you always wear this, or is this like when you're doing a sleep test? Okay.
Yeah, it's like you just get monitored for 24 hours because they might probably only if there was some sign that things aren't ideal.
Yeah.
And they'd be like, you're sort of at the edge of the range. Let's wear this for 24 hours.
Is the windshield wipers real?
Sam, windshield wipers are real. They go on cars, but
Okay,
there isn't a good way to break up the floaters that you have in your eyes.
Okay. Ashley, do you have a science background, or are you just a science genius?
Oh, I wouldn't call me a genius, but I just have been super into science for like a hot minute. Fun fact, mostly because of Crash Course. I've just been really kind of into science, and it's just been one of those things where the internet is vast and you have access to so much knowledge.
That's
so sometimes
awesome.
I go on deep dives.
Doesn't bode well that you just beat the main editor of Crash Course at a science game.
True.
I did nothing of the sort. I'm just over here.
I edit the content. I don't need to,
You don't need to know
generate it.
until you
I
do it.
hire experts to generate
Yeah.
it. And I've been deep in organic chemistry, which is not
Any of
cyborg related
what
at
we've
all.
done.
I could tell
Yeah.
you about aldol and Claisen reactions. I can push electrons around. I've basically spent 2 years taking 2 semesters of organic chemistry, and I'm ready to take the MCAT now.
Oh my God. And never again will you have to think about an aldol. But thank you for that very important work. It has not, as we say, helped you. And you have been punished for not snapping for my poem with 0 Hank bucks at the end of the episode, whereas Ashley and Sam have tied for the win.
Hey, see, that's what we get for snapping for the poem. That's what it is.
That's how it works. Now it's time for Ask the Science Couch, where we've got listener questions for our couch of finely honed scientific minds. This question is from @GiantBonsai. How is the mind able to send pulses to technology to control it like our actual body in the parts that require conscious voluntary control? How do the signals know where to go? So this is a thing that we're seeing now with some prosthetics, where you can actually think about moving a prosthetic and the prosthetic will move, which is wild and new to my life. Like, it did not exist when I was a teenager and now does. I don't know the answer to this question, though. Ceri, do you have any idea?
I do. I'm being punished by having to do more work for this podcast, as always. But yeah, so there are multiple kinds of prosthetics, like you said. Some are just cosmetic, like filling in the missing space on the body. Some of them are muscle controlled and provide structural support through like a harness or cables attached. And then now we are venturing into the expensive myoelectric prosthetics. Myo is like from muscle, and then electric is electric, that work together with the electrical impulses from the nervous system and like the muscular system, and have rechargeable batteries and other electronic components. And basically what it does is it takes advantage of the fact that our bodies are powered by electricity, and our muscles already use electricity to move. Neurons and muscle cells can use membrane potentials to generate electrical signals. They generally, at resting state, have a negative charge inside and a positive charge outside. And then when they want to send an electrical signal, or when your brain tries to send an electrical signal, then sodium channels, or like different ion channels, open so that the charges can rush in. And then when they reach a certain threshold, that creates an electrical impulse. And so neurons signal other neurons, and then neurons can also signal muscle cells. And that's what causes the muscle cells to start contracting. And in myoelectric prostheses, it's not as simple as like changing a light fixture. So our bodies don't have wires, and we don't understand the nervous system that much. So like in the way that you know what you're going to see when you pull a light off the wall, like you'll have a ground, you'll have a positive and a negative, and maybe like a fan cable. your arm doesn't have those hookups. So it's like, you don't have like this finger nerve goes here. We know where the muscles are, but when we're building a prosthesis that hooks up electrically, there are electrodes that sit over working muscles or active nerve areas that still do generate that electrical impulse. And that changes from person to person because different people have different kinds of nerve damage and different working areas of their body, so that when they move certain muscles in a certain way, and like they try and place the electrodes and program the response to the arm or leg or whatever limb is being replaced in such a way that when they flex certain muscles that mimic what they would have done with a biological limb, the microprocessor and the motors in the electronic prosthesis mirror that. If, for example, you had a prosthetic arm and you flexed a muscle that you typically would to clench your hand, then that would send a signal to the robot arm to clench. And it's very difficult to do, and a lot of them don't work very well, and people don't trust them super well because it's like everyone's body is different, and there's often like a really delayed reaction time where like if you think about clenching your hand, it's almost instantaneous. And when that doesn't happen instantaneously with a prosthetic, that becomes really discouraging for folks who are doing these like motor tests. And so there's a whole bunch of literature, not just about how they work, but how they work not well enough to be mass produced or like very usable for people because they get frustrated or like feel out of sync with it.
To me, that seems like it might be like a pretty hard problem to overcome as well. It's just hard to, it's the nervous system is not electronics. And so that interface where you've got this like cool metal arm, like forearm sticking out of your elbow, does seem a bit of a ways away. How does the nerve impulse get through this skin? Is it just very close to the skin?
Yeah, I think it's very close to the skin in the way that you can kind of like with a defibrillator, you can put patches on the outside of the skin and restart a heart. Sure. I think the skin is conductive enough to transmit the electrical signal or transmit enough of an electrical signal to be read.
Yeah, because in like, science fiction comic book Cyborgs, it's just like the flesh just fuses into, but that is not how this works. We have to contain the body. Has to stay on the inside. Otherwise, there's going to be a lot of issues.
Yeah.
I think that part is definitely, I don't know, we've like sort of explored that territory, and I didn't research pacemakers and things like that. Like there is separate research into technology that integrates with organs safely. But it's not necessarily controlled. It like can help control the electrical impulse of the organ. So like a pacemaker helps you regulate your heartbeat, but is not in turn controlled by your heart. Like you're not thinking about it. It's just a battery that is ticking along.
One day at a time, science. I have faith in you. But the bad news is I will die. Probably before you finish that job.
Wow.
Put your brain in a jar and then put wheels on it, and then it'll drive toward the light.
Yeah, there you go.
If you want to ask the Science Couch your question, you can follow us on Twitter at SciShow Tangents, where we'll tweet out topics for upcoming episodes every week. Thank you to @Judah Kraz, @Mala Monster, and everybody else who tweeted us your questions for this episode. Ashley, thank you so much for joining us. If you want more Ashley Roboto, where can they go for that?
Thank you so much for having me. It was an absolute pleasure to be here. Oh my gosh, I am just buzzing with excitement that I was even here with y'all. It was a great time.
Super fun.
If y'all want more of my content, I stream Monday, Tuesday, Thursday, and Friday on Twitch at twitch.tv/Ashley Roboto, and just @Ashley Roboto is where you can find me anywhere. Twitter, TikTok, Instagram, all those. I'm everywhere.
Thank you so much, Ashley. If you like this show and you want to help us out, it's super easy to do that. You could subscribe on your podcast app of choice. If you're new and you are just coming to listen to Ashley, you can also go to patreon.com/SciShow Tangents and become a patron and get access to things like our newsletter and our bonus episodes of our show. Second, you can leave us a review wherever you listen. That helps us know what you like about the show and helps other people know how great we are. And finally, if you want to show your love for SciShow Tangents, just tell people about us. Thank you for joining us. I've been Hank Green.
I've been Ceri Riley.
I've been Sam Schultz.
I've been Ashley Roboto. Hi.
SciShow Tangents is created by all of us and produced by Caitlin Hoffmeister and Sam Schultz, who edits a lot of these episodes. Our social media organizer is Paula Garcia Prieto. Our editorial assistant is Deboki Chakravarti. Our sound design is by Joseph Tuna Medish, and we couldn't make any of this without our patrons on Patreon. Thank you. And remember, the mind is not a vessel to be filled, but a fire to be lighted.
But one more thing. In the mid-2000s, Ged Galvin of Yorkshire, England, was in a terrible motorcycle accident in which he suffered severe internal injuries. As a result of the accident, he needed to use a colostomy bag. But in 2009, he underwent an experimental surgery to build him a new colon. Ooh. Doctors removed a muscle from his leg, molded it into a sphincter, and filled it with remote control electrodes that allowed his sphincter to open and close with the push of a button on a cell phone-sized remote control that he carries with him.
Oh my God.
And since he is British, many of the articles about Ged's new sphincter call it a bionic bottom.
So polite.
Holy, what? Why don't I know about that?
Button operated too. It's just like, I'm no straining to poop. Just, I want to poop now. Open that chute.
Welcome to SciShow Tangents. It's the lightly competitive science knowledge showcase. I'm your host, Hank Green. Joining me this week, as always, is our science expert, Ceri Riley.
Hello.
And our resident everyman, Sam Schultz.
Hello.
But you know what Sam Schultz isn't ignorant of?
Oh, no. Don't do this to me.
The Vampire Diaries. Can you explain to me how the vampires work in the Vampire Diaries?
Well, they at the beginning of the show, in the first season,
like normal
of the
vampires,
show,
can they go out in the sunlight?
They need a magic ring to go out in the sunlight.
Grain?
So no, a magic ring. A ring.
A ring.
You know, they eat a piece of toast.
Yeah.
Yeah, they need a magic grain. That would actually be pretty cool.
It's quinoa.
So yeah, when you first meet Stefan Salvatore, the main vampire of the show, he's got this big ugly ring on.
Oh, he can go outside.
That's what he needs to go outside. It's a daylight ring, and a witch has to make it for you, and eventually everybody just has one.
Oh, so okay. It's not just like one. You can get more. So no, it's not like the Highlander where you have to like stab Stefan Salvatore to get the ring.
Well, in the first season, that is kind of the way it is. Okay. There's only two, and it's like, oh, we need these rings. But then like the next few seasons, it's just like, oh yeah, my necklace is
So
one.
they're just people who drink blood now.
Yeah.
Do they like garlic?
Yeah, they can eat garlic. That's a very specific. There's a scene where Stefan's cutting garlic, and he's like, I love Italian food.
So that was made up.
Yeah.
You disregard that piece of the
They can't
lore.
turn into bats, which is really dumb. I think they should be able to turn into bats. I think that's like the coolest vampire power, I think.
Yeah, but it's really hard. I understand turning into a cloud of bats, but turning into one bat, that's just against all
it's hard
science.
scientifically. I thought you meant like the graphics would be hard to do.
Yeah, it's just conservation of matter is kind of a big deal.
Give you one really heavy bat.
Yeah, that's right.
Yeah, a large bat.
It's just all the protein. It's like a neutron star inside of the bat.
Yeah, infinitely dense bat.
It's like, yeah. Perfect. You've solved the problem.
Yeah. Any other questions?
Yeah. So they do drink blood.
Yeah.
Do they kill people?
Oh, all the time. There's some of them that drink blood so hard that they make the head pop off of the person whose blood they're drinking.
That's not how that works.
They're called rippers.
They suck so hard that it just goes whoop. Or do they bite into?
Who really could say? But they grab a person, they bite, and then at the end of the biting, their head just pops right off their body. It's really cool, actually. But they're bad vampires. We don't like them.
Because they make the heads pop off instead of just killing.
There are good and bad So is there are there some like Lestat who just like stumble around the streets of Paris, France, and chew the heads off of rats?
Oh yeah, that's Stefan. He's like that. Except he's secretly a ripper, and he can't control himself. So he has to drink animal blood because if he gets one drop of human blood, he'll go nuts and he'll start popping people's heads off.
Does he pop animals' heads off?
No, he can drink that stuff normal. He could like meet a cow.
It's not
and
like
be
good
The
stuff,
idea
yeah.
of just like mice with just pew, 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 pew,
Yeah.
pew, pew, pew.
Yeah, you're shotgunning mice and their heads are just flying out there.
Yeah, these are all better ideas than the Vampire Diary Explorers.
Is it like the Princess Diaries?
No, they write in a diary like one time in the first episode, and then
There's
that's
no
just
diaries
it.
after that.
There's a few, and every like once a season, somebody will write in
Dear
a diary.
diary, I love garlic. Pop, pop.
Yeah. Why are you asking me about this? To make me embarrassed?
No, you seem to know a lot. I feel like I should be aware of how it works.
It's a good show.
It's an everyman's sort of knowledge, I think.
Then there's werewolves too. We haven't even got into the werewolves.
Do they also drink blood?
No,
Do
eat
they also
food.
pop heads off?
They could if they wanted to. Then there's half wolf, half vampire. They can do all.
What?
They can do all of the above.
Whoa.
Okay.
Yeah.
Are there witch, wolf, vampire?
No, you can't be You can't be magical and be a vampire or a
No,
werewolf.
Oh, that's
at
a
the same
rule.
time.
That's a real rule.
Yeah, if you
Okay.
get turned into one of those, you lose your magic. Except Stefan's mom.
There's always one.
Spoilers. Okay.
Good warm up for Halloween, guys. We're getting in the
just
spirit.
getting ready. So every week here on SciShow Tangents, we get together to try to one-up, amaze, and delight each other with science facts and Vampire Diaries facts, while also trying to stay on topic. Our panelists are playing for glory and for Hank Bucks, and I'll be awarding those as we play. So at the end of the episode, one of them can be crowned the winner. But as always, we got to introduce this week's topic with the traditional science poem. This week, it's from me.
Oh, a treat.
A treat for the people who aren't watching on video. You only know that the treat made a number of noises.
Yeah.
A thing we've known through modernity. Atoms are unchanged for eternity, even when they change their chemicals. An oxygen is oxygen, and hydrogen is hydrogen. Always the same, they never change. Whoa, but then they do. Big atoms can decay from one element to another cascade. Particles emitted, they're emitted. Particles emitted, they're emitted. Whoa, oh, they're radioactive, radioactive. Whoa, oh, they're radioactive, radioactive. No one tell Imagine Dragons. We didn't get the
Please
rights to
don't
the
tell
song.
on us. We'll get in trouble.
You're allowed to say that was really cool, but you have to be kind of secretive about it. On Twitter.
Yeah. When you're tweeting about it later.
The topic for the day is radioactive. Specifically, I could have kept going because there's other kinds of radioactive, I feel like.
You're saving that for your new album, right?
But I gotta save it for the new
I think you committed a crime that Weird Al doesn't commit, where Weird Al changes the chorus. I think that's where you're gonna get in trouble.
I changed the chorus.
What is it?
They're, not I'm, they're radioactive.
Oh,
See?
Okay, we're fine. We're fine.
Yeah.
I see.
So our topic for the day is radioactive. Ceri, what does radioactive mean? Did I get everything in my 85-word song?
I mean, you got the gist of it, which is all we can ever hope to do with this definition section. So, radioactivity is just radiation emitted by a radioactive material, and that can be lots of different forms. It can be alpha particles, beta particles, neutrons.
These are specific things.
Yeah.
One of those things is like a high energy electron, maybe, or photon, or what are they?
Beta particles are electrons that are not attached to atoms, so like spitting out an electron. Alpha particles are the chunky ones, so they're 2 protons, 2 neutrons.
Oh, so big boys.
Big, large boys.
That's basically a nucleus at that point.
Yes, basically like a little bigger than that's like a helium-ish size.
Yeah, it's a
thing.
helium nucleus.
That's an alpha particle, the giant one. Beta particles are like spewing out a little electron. The third is a neutron, which is like a hydrogen, kind of. It's a little smaller, but spewing that out. And then there's electromagnetic radiation, which is like X-rays, gamma rays, visible light.
And that's just like light. Yeah.
Light, yeah.
Light and different. But gamma rays are like light that'll do you a damage.
Yeah, there's light that will not do you damage, and then there is light that is high energy enough that
It'll get in there.
Make you into
get
a Hulk.
into atoms and molecules, and start messing things up and like.
So radioactivity, is it pew, 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 shooting? Is that what radioactivity is? Something shooting off of something? I read a lot about radioactivity. I couldn't make heads or tails of it.
Oh, wow.
Especially nuclear energy.
So there's the radiation.
Okay.
This is the confusing thing. There's radiation that comes off of radioactive things. And something that's radioactive is, I think all it's really saying is that like there are waves, there's emissions coming off of this thing. But I think that in general, when we say radioactive, we mean things that are emitting particles, like actual pieces of themselves. And when that happens, they can actually decay into other elements.
Okay. Can we talk about how you said, oh, wow, after I said I read about it and couldn't make heads or tails of it?
Look, you know about Vampire Diaries.
You said the thing I said.
No,
I was
wasn't-
right.
It's not. Yeah, it's not. I wasn't oh-wowing you. I was oh-wowing the like terminology mess that we got ourselves into because we didn't really understand what was going on when we first started naming this stuff.
Okay.
Yes.
Oh,
yeah.
interesting.
That is the big problem. It's like depends on where you draw the circle. Because technically, if you wanted to be stubborn about it, you could claim that
that like
so
my
many
LED is radioactive
your light
because
bulb
it's emitting lights.
is
A
emitting
light bulb's radioactive because it's okay. I see.
Yeah.
I see.
Well, because everything, to some extent, or a lot of things, are emitting small doses of radiation. So, like,
And there's like a background radiation everywhere.
yeah, there's
That's
cosmic
yeah,
radiation
absolutely.
in the universe.
I'm radioactive both because I have potassium in me that has some unstable potassium, a little bit of unstable, but also because I emit infrared light because I'm warm.
That's nice. So where does this word come from that we're so confused about?
Yeah, radioactivity. So I looked up radioactivity, but I assume radioactive is similar. It's fairly recent, which is probably why we're so confused about it, because radioactivity comes from the French radioactivité, which I think is like,
the same word, yeah.
which was made up by Pierre and Marie Curie in 1898. They were like,
Oh.
what is this new element? They had some predecessors who had studied uranium and the fact that it emits radioactive particles, but they were the ones to first use radioactivity as an adjective or a noun. Whatever part, radioactive
Whichever
as an
one.
adjective.
This is a science
radioactivity
show.
as a
This
noun.
isn't a grammar show.
To describe what they were working on in uranium and polonium and radium. And they were like, radioactivity is a property that these things have. And further back than that, I tried to trace it. There are several different ways that we started incorporating like radio into words, and especially in recent history where like radio means the machine, the radio, and then we tack that onto words. But at this point, the use of radio in radioactivity came from the Latin radius, meaning ray.
Oh, yeah.
And so it came more from the idea of a ray of light, a beam of light, and the emission property of it to get into that, like the radiantness of it, as opposed to the straight rod in the middle of a circle.
It's a little bit frustrating to me that light is radiation. Now, I know that that's ridiculous, but it does seem like we got this word that turned bad. You know, at first it was like, this is interesting, and then it was like, this is cool. This is good. We should shine it into our eyeballs. And then I was like, actually, it's terrible. We should be terrified of it. And that has lasted a long time. And now people sort of like radiation is synonymous with ionizing, dangerous, cancer-causing radiation. Whereas it's sort of important to realize we also get to, like, people use that word for visible light.
It was going to be a Halloween topic, but then I moved it up a little bit because it's scary.
Yeah, you could definitely make a Hulk.
A giant
with it.
A giant ant, a giant mantis, giant octopus.
Spider.
Really take
You
your
could be a
pick.
spider
Giant
person.
spider. Yeah.
It's going to be time to go into the quiz portion of our show where we're going to be playing radioactivity, this or that. So our bodies are constantly interacting with radiation that's coming from space, from materials found on Earth, from things that we make. And the National Council on Radiation Protection and Measurements estimates that the average person in the U.S. experiences around 6.2 millisieverts per year. So about half of that is from natural background radiation, and the other half is from man-made things like medical stuff, industrial sources. Fortunately, this amount of radiation does not seem to do us very much harm. Researchers have measured the radiation doses we experience from a variety of sources. So today, we're going to be playing This or That Radioactive Edition, where I will present to you two things, and you will have to guess which one is more radioactive. Ooh. Radioactive.
Whoa.
Whoa. So first, we're going to start out with medicine. Which one of the following things requires a larger dose of radiation? An X-ray of your chest or a CT scan of your chest?
Couldn't tell you what a CT scan is exactly.
A CAT scan. That's what it's a CAT scan.
Okay. Now I know at least what the word is, but I'm still not really sure what that does.
Yeah. That's understandable. You're not old enough to need to know all the medical things that happen.
Your body hasn't broken down yet.
Yeah. I just read a biography of Antoine Lavoisier, the guy who basically sort of invented the modern concepts of how chemistry works. And as he was in his little cell getting ready to be put to death during the French Revolution, he wrote a letter to his cousin, and it said something to the effect of, "At least I have been spared the indignities of old age."
That's nice. He was putting a good spin on it.
Yeah. He was like, getting old kind of sounds like it's awful hard. Anyway,
I
don't want to have to learn what a CT scan is.
Yeah. All right. I guess it's probably CT scan, because X-ray I feel like it's like, this is the trick. It's a trick question. Basically.
That's what I think too. I think it's a CT scan because I think it takes longer. I feel like an X-ray, you just go, bleh. You get X-rays at the dentist. Even your chest, you could probably do it pretty fast.
Well, you are correct, and all the people who know about medicine were screaming the answer to you, because CT scans are actually just a bunch of X-rays.
Oh,
yeah.
So
okay.
you take X-ray images from a lot of different angles, and that requires obviously a larger radiation dose than a single X-ray would.
Oh, that's fun.
So they both produce ionizing radiation that involves high energy wavelengths that allow the particles to penetrate the tissue, and a chest X-ray has an average radioactive dose of 0.1 millisieverts. A CT scan of the chest has an average radiation dose of 7 millisieverts.
And we get 6 per year normally? Is that what you said?
We got 6.2 a year. That's right.
Okay.
So yeah, you only get a CT scan if you need one.
Okay.
That's for sure.
It's like 70 little chest X-rays going pew, 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 pew.
And the radiation that doesn't get absorbed in these things, that's the stuff that makes it through and produces the final image. And the doctors use these effective dose values to understand the risk that the procedures might pose to the body overall, and to balance that with the potential benefits of using these scans. Round number 2. We've moved from medicine to travel. Which of the following exposes airline passengers and crew to more radiation? A flight from Frankfurt to San Francisco, or a flight from Hong Kong to Hartford?
Hong Kong to Hartford? Gosh. Frankfurt to San Francisco. I feel like the poles have something to, maybe like, I don't know. You're going over the top or something. I'm
going to guess Hong Kong to San Francisco because...
You can't. You got to guess Hong Kong to Hartford.
Hong Kong to Hartford. I'm going to guess Hong Kong to Hartford because just to, I can't logic through it. This is beyond what I can do. So the best I can do is guess.
So as you have correctly surmised, space is a source of radiation. There's X-rays, there's high energy particles, there's gamma rays, and they also react with our atmosphere and create secondary radiation that can reach us. And while you fly, you are at a higher altitude, and that means you're less shielded from all that radiation. There are other factors that impact the amount of radiation that flyers experience as well, like the duration of their flight, of course, and also the distance from the equator, which makes a difference because of how Earth's magnetic field pushes radiation towards the poles.
Good job, Sam.
You were right, but you still got the wrong answer.
Well, I wasn't sure where any of those places were exactly, so that was a problem.
So, in 2016, researchers used models of solar activity and its effect on the energy of particles impacting the Earth to estimate the average radiation doses that different passengers would experience on different flight routes. And with their model, they calculated that on average, a passenger from Frankfurt to San Francisco experiences an average dose of 70.7 microsieverts, not millisieverts, so even smaller, while a passenger from Hong Kong to Hartford experiences 93.2 microsieverts. And in general, they found that the highest dosage flights were ultra long haul flights between US and Asia, a lot of those, and above the halfway point between the equator and the North Pole. I'm surprised that you could go from Hong Kong to Hartford. It's Hartford.
Yeah.
No offense to Hartford.
The Hartford International Airport, you know.
Yeah. Can you really do that? We can go to like 4 places from Missoula.
Yeah.
They can go to Hong Kong.
That doesn't seem fair.
It doesn't seem fair at all. And our last section of This or That, it's the food edition. Which one of the following has more radiation? 1 kilogram of beer or 1 kilogram of bananas?
Beer or bananas? Beer, I don't know. We talked about like whiskey barrels having the nuclear bomb radiation in them or whatever. So I'll just go with beer. Do you put beer in barrels? I don't know.
No.
You don't? Some people must. I feel like I watched an old Popeye
I'm sure they used to.
I feel like I watched a Popeye cartoon where beer was in a barrel.
With the XX whatever on it, and it lugs it out of the barrel.
Or isn't that what Dumbo's drinking out of barrels or something like that?
No.
Well, what do you think?
I think it's bananas because of the potassium, but I don't know what else is in beer. Beer is like a lot of carbs and water, and I don't think that's very radioactive.
Ceri's right.
Damn,
It's a lot of carbs and water. But all of our foods to some degree have some radioactivity, in part because of carbon isotopes, and there's going to be carbon in anything you eat. But there's also other elements like potassium-40 or radium-226. The units used to report these values are in picocuries per kilogram, where a picocurie describes the amount of ionizing radiation released when an element goes through radioactive decay and emits energy. So in the case of bananas versus beer, bananas have around 3,520 picocuries per kilogram, and beer has 390. Luckily, the amount of radiation from a banana doesn't really add up to very much when we eat them. However, the radioactivity of bananas has inspired an unofficial radioactivity scale called the banana equivalent dose to describe the radiation exposure in terms of bananas, where 1 BED equals 0.1 microsieverts.
Scientists love having fun.
That's right. All right, Sam, you have 1 point, and Ceri has 3 going into the break. Next, we're going to take a short break, and then it'll be time for the fact off. Welcome back, everybody. It's time for the fact off. Our panelists have all brought science facts to present in an attempt to blow my mind, and after they have presented their facts, I will judge and award them Hank Bucks any way I see fit. To decide who goes first, I have a trivia question. The fungus Cladosporium sphaerospermum seems to thrive in the presence of radiation. So naturally, research sent a sample to space, monitoring it for 30 days aboard the International Space Station. Their experiments demonstrated that a 1.7 millimeter thick bed of fungus could lower radiation levels in the area by 2.17% compared to an area that wasn't shielded by the fungi. Based on their results, the researchers estimated how thick of a layer of Cladosporium they would need to create Earth-like conditions on Mars. How thick would this fungus blockade need to be?
What units do you want?
Let's do feet.
Oh, feet.
Feet.
Oh, sorry. I gave something away.
That's big. Yeah, I was gonna get it wrong by.
Also, meters would be fine.
Okay, I'm imagining swaddling myself in a blanket of fungus. What would make me feel safe on Mars?
I think 20 feet.
Oh, that's a lot. Okay, I'm gonna say 2 meters.
It's just dang mushrooms.
Ceri says 2 meters. It's 2.3 meters.
Wow.
My mind palace did me well.
Just picturing being enveloped by 2 meters of fungus on every side.
Yeah, and do I feel safe now? Yes. The
I feel
answer
safe
is yes.
now. That's the only way to feel safe. Just as long as you have a snorkel.
Yeah, yes,
Right.
a straw to breathe out.
So that means you get to decide who goes first.
Sam, you can go first.
Oh, no.
I'm taking the
Okay.
coward's way out.
I wasn't prepared for this. The Boy Scouts of America, just for those of you out there who don't know, is a youth organization devoted to teaching kids junk like tying knots, catching fish, identifying plants, whittling, good forest-based activities that all blue-blooded American children ought to know. When you get good enough at these things and pass some tests, you get a little badge to put on your sash, and those with many badges are most esteemed among Boy Scouts. But in addition to your more traditionally outdoorsy array of badges, there are some weird-ass badges, like dentistry or fingerprinting, or one that indirectly led to a Michigan Scout's backyard being declared a Superfund site, atomic energy. David Hahn, a Boy Scout, was awarded the Merit Badge in Atomic Energy in 1991 after completing some rudimentary tests and projects like building a model of a nuclear reactor with cans and straws and household items. But David Hahn was also a naturally born scientist and had been studying chemistry since he was 10, inspired by the Golden Book of Chemistry Experiments, which was given to him by his grandpa. And he had spent the intervening 4 years doing things like learning how to make nitroglycerin and blowing up his bedroom numerous times, to the point where he had been exiled to his backyard as kind of like his little laboratory where he was doing his chemistry experiments. So David, at this point, was a bit drunk off the power of chemistry and decided that he didn't just need to leave his exploration of nuclear power at a mere model. So he started sending out letters to various nuclear energy organizations, including the Nuclear Regulatory Commission, posing as a high school teacher and asking for educational material to use in his classes, like where in the natural and man-made world one could find radioactive material. And they sent him all kinds of info back, enough that he eventually figured out that he could create purified thorium out of parts of gas lanterns combined with lithium from $1,000 worth of batteries. So he just poured lithium on this. Then he put a Geiger counter on his car's dashboard and drove around town, finding all of the radium he could from old watch faces and instrument panels, and used that to irradiate his thorium, which he then combined with americium. Oh, shoot. I wasn't even close. Which is what's in smoke detectors that makes them detect smoke. It's radioactive as well. And he built a breeder reactor, which I don't entirely understand what a breeder reactor is or how it works, but it's a reactor that can somehow make more fissile material than it uses. And they were a major source of interest of nuclear scientists in the 50s, but a couple of the major experimental breeder reactors ended in near meltdown, so they lost popularity. But David made one, and it worked. But it started working too well, so well that radiation was becoming detectable up and down his block, so he took the reactor apart and hid it in his car trunk. But in a case of weird mistaken identity, a cop ended up searching his trunk for something not even related to him, basically, and ran across the reactor and started messing with it. And David said, "I wouldn't do that. That's radioactive." Then, a few days later, flash forward a few days later, the FBI, the EPA, and the NRC are in David's backyard, detecting background radiation 50 times higher than normal background radiation. And ultimately, his shed was declared a Superfund site, dismantled, sealed in barrels, and buried in the Utah desert alongside material from nuclear bomb testing. And all of this after he had already gotten his merit badge. So the lesson is, as story editor Alex Billow put it when I sent him this story, there is such thing as trying too hard.
Oh, don't I know it? That's amazing. I'm not sure what my favorite part is. The part where he drove around with a Geiger counter on his dashboard so he could find radium watch faces?
Yeah,
and apparently that worked?
That seemed like it was a bit of an ostentatious, move on his part, but it actually accidentally did end up working out because he passed an antique store and it went crazy. And then he found an old clock, and inside of the old clock, he found a vial of radium that had been left so he could touch up the clock face.
Oh, wow.
And that was what he used mostly to shoot his thorium with.
But I think probably the best part is that they buried his shed in the desert.
I think that's my favorite part too. They put it in barrels and took it away and put it in the desert.
I like the email or the sending letters to the government, pretending to be a teacher.
That is also very good.
All right, Ceri, what do you have to compete with Nuclear Boy?
So certain radioactive isotopes of elements have an ominous nickname. They're bone seekers. They tend to accumulate in our bones if they get into the human body somehow. So, for example, strontium-90 is in the same periodic table group as calcium, so one of the columns, and acts chemically similar to it. This radioactive form has 38 protons, like a stable strontium atom, but what makes it an isotope is its whopping 52 neutrons. And in the late 1950s and early 1960s, as nuclear testing was ramping up and isotopes like strontium-90 were getting released into the environment as waste products, there was a lot of curiosity about how much people were getting affected by them. What's happening here? So the physician, Louise Rice, along with her husband and some other scientists, mostly environmental scientists, led a project where she was basically the tooth fairy, but in a scientific way. And by that,
that's a great. I was like, are they gonna grave rob?
No,
are they gonna grave rob?
It's much
that's not
cuter
allowed.
than grave robbing. And by that I mean her team collected and analyzed thousands of baby teeth from kids in the St. Louis, Missouri area in the U.S. for their strontium-90 content because
Why
it's a bone
not?
seeker.
They're around.
Yeah.
There's plenty of teeth.
We're not doing anything with all these teeth.
Yeah, why keep them? That's creepy.
It's so much easier than grave
They just
robbing,
gotta
surgically
get in
taking
touch
out
with the
bone.
tooth fairy and be like, here's a grant.
And so in a massive public outreach effort, Rice's team visited schools and other community centers and explained to families how strontium-90 from nuclear tests could make its way into human bones by being sprinkled into water or dairy products and then making its way into food that the kids are eating. And they talk
And then it seeks your bones.
Yeah, it seeks your bones and gets in there and just makes a little home. Swaddling itself up in the rest of the bone, like me and the fungus. And they talked to caregivers for consent and spread scientific literacy, which is honestly very exciting to be able to say about an old science experiment. Thrilling. Seems ethically sound. And families that wanted to participate sent in geographic information about where they've lived, their kids' baby teeth, and in return got a fun little button that said, I gave my tooth to science, which in my nerdy opinion is much more fun than a couple of quarters.
Well, get both, ideally.
After the first 2 years of analysis on over 65,000 teeth,
Whoa!
Rice published a paper on November 24th, 1961 in the journal Science and reported that they did find elevated levels of strontium-90, and therefore the stuff getting into the environment from nuclear tests was also getting into humans through children, but also adults, presumably. And this paper, along with some testimony from her husband in front of the Senate, helped influence the U.S. government to sign on to the Partial Nuclear Test Ban Treaty in 1963.
That's
but
awesome.
There was still plenty of tooth research to do. This team kept collecting baby teeth from St. Louis kids through 1970 and ended up with around 325,000 teeth in total.
Wow.
And further analysis led to findings like children born in 1963 had around 50 times as much strontium-90 as children born in 1950, which was before many, but not all nuclear tests. And so there was a pretty big difference. And the whole using teeth to study radioactivity thing stuck around, as later studies showed that strontium-90 in baby teeth decreased by around half in kids born in 1968 after the treaty had been put into effect. And so we had this like peak around peak nuclear testing, and then the governmental policies, environmental policies really did help people be less radioactive. And even nowadays, the Baby Tooth Survey inspires other initiatives to detect various kinds of pollution that can end up in bones and impact human health. And it's very cool to me that this weird, wonderful collaboration between scientists and the public and a scary amount of teeth existed, and people were excited about it.
That's wild. Hundreds of thousands of teeth is a significant haul if you get them to the tooth fairy.
Yeah.
And it makes me wonder, like,
Fraud.
is that part of the deal? Like, somebody's got to be funding the research. Is it the tooth fairy?
Big tooth.
Could be. Tooth Fairy is very interested in this niche field of research.
I like the idea that there's a bunch of tooth fairies and a lot of them are like independent like operators. They have small businesses, but then there's like one that's like sort of rolled them all up. It's like a corporate. It's called Big Tooth.
Yes.
Gosh, I thought Sam had it in the bag.
Yeah, this one's pretty good though, and she got 3 points. So
let's just say that Sam got 100 points in the first section, and Ceri got 300 points in the first section.
Oh, okay.
I just feel like I need more decimal places to work with here. And for these facts, I think that Ceri's was probably like a 500-point fact, and Sam's was like a 450-point fact, which are still both are very high. But
I got more than Sam in both.
Yeah.
That leads me to believe that Ceri is the winner of the episode.
Wow.
They ground up hundreds of thousands of teeth.
I also have never really reckoned with just how many teeth are out there. Like, this was 12 years of actively collecting with people's consent.
It's not. It's by far not anywhere near the majority of the teeth.
No.
It's a tiny fraction of the teeth.
We're fighting about sand. Tooth as a renewable resource. We keep making babies. We keep making teeth.
Yeah.
Someone needs to start communicating with the tooth fairy about these grants, about the research we can do.
Tooth sand? Is that what you're suggesting?
I don't know.
We're gonna make concrete with it.
Yeah.
Oh.
Look, if this is your first episode at SciShow Tangents, this is it. Congratulations. Thank you so much. And if you're wondering which episode to send to a friend, and you're like, well, I'd like more people to know, this is the one.
Yeah.
This is the one. This is the one that put you over the edge. You're like, it's got a Imagine Dragons song in it. The first part's all about Vampire Diaries for some reason. Both of the fact-off facts were just totally unhinged and amazing human endeavors. And you're not even done yet, because now it's time to Ask the Science Couch, where we ask a listener question to our finely honed virtual couch of scientific minds.
James on Discord asks, how did we determine fatal dosages of radiation other than error? I'm hoping nobody trialed that.
I think most of it is from people who have died from radiation exposure. So it's not like intentional trials in as much as like war is an intentional trial. But like one of the biggest research institutes for it is the Radiation Effects Research Foundation, the RERF, which is a collaboration between Japan and the United States. And a lot of what we know about lethal doses and cancer frequencies and stuff has been by studying long-term health records of the survivors of the bombing of Hiroshima and Nagasaki. So that, at least in English language, is where most of what we know comes from, as far as like risk to cancer, exposure to different amounts of radiation, LD50. We study with radiation, it's specifically LD50/30 is the figure I found more than once. I don't know, which is the dose of radiation expected to cause death to 50% of an exposed population within 30 days, because radiation sickness has to set in. It's not like an immediate vaporization at the amounts that we have on Earth.
That makes sense. And the ability of people to take the opportunity to continue to learn is pretty... it seems like the right thing, especially because like it's not like we are done with radiation on the planet. So, and also like it's a super important thing when it comes to space travel. Like it's just
Right.
I guess you could swaddle yourself in fungus, but we don't have solutions to all of those problems yet. If you want to ask the Science Couch your question, you can follow us on Twitter at SciShow Tangents, where we'll tweet out topics for upcoming episodes every week, or you can join the SciShow Tangents Patreon and ask us on Discord. Thank you to Buns on Discord, at Boy with Headache, and everybody else who asked us your questions for this episode. Did you laugh at the way I said Buns?
No, I just laughed because of buns. Thank you, buns.
Both at the way you said it and the name Buns.
If you like this show and you want to help us out, it's super easy to do that. First, you can go to patreon.com/SciShowTangents to become a patron and get access to things like our newsletter and our silly little bonus episodes. Second, you can leave us a review wherever you listen. That helps us know what you like about the show, and also it helps other people learn about the show. And finally, if you want to show your love for SciShow Tangents, just tell people about us. Thank you for joining us. I've been Hank Green.
I've been Ceri Riley.
And I've been Sam Schultz.
SciShow Tangents is created by all of us and produced by Sam Schultz. Our editor is Seth Glicksman. Our story editor is Alex Billow. Our social media organizer is Julia Buzz Bazzaio. Our editorial assistant is Deboki Chakravarti. Our sound design is by Joseph Tuna Medish. Our executive producers are Caitlin Hoffmeister and me, Hank Green. And we couldn't make any of this without our patrons on Patreon, of course. Thank you. And remember, the mind is not a vessel to be filled, but a fire to be lighted. One more thing. The Hanford site in the U.S. churned out radioactive plutonium during the 1940s, during the Manhattan Project, and is now a huge government cleanup project. Ongoing efforts involve searching for radioactivity with detectors mounted on helicopters so they can monitor for any unexpected leaks and waste can be disposed of safely. Now, rabbits or other small wildlife can burrow into contaminated areas without realizing it and lick up the radioactive salts. And because what goes in must come out, they leave radioactive poop piles across eastern Washington. And that poop is radioactive enough to get picked up by these detectors and become one more thing that needs to be cleaned up. Although it is apparently not a top priority.
Just looking for the big spills, and then it's a little rat. Ah, man, Jim, it's another rabbit poop. It's another freaking rabbit poop.